I think yesterday we were actually discussing about the protein metabolism. And if you people remember, I told you the overview of the protein metabolism here. Yes. In this overview, I told you that after consuming the protein, the protein will undergo digestion to form amino acid, and that amino acid will be absorbed. And after absorption of the amino acid, what is going to happen? The amino group will be actually removed in the form of ammonia. And ammonia being very toxic there, we have to actually detoxify ammonia, and that is going to the liver and getting converted to what's it? Urea. But now the first challenge is actually transporting this ammonia itself, and that is known as what's a transamination. इसको हम ना transamination कहते हैं सर, ठीक है ना? Now in transamination, I told you already yesterday that ammonia is transferred from many tissues to the liver in the form of glutamine, and ammonia is transferred from the brain to the liver in the form of glutamine only. But from the skeletal muscle to the liver, it will be transported in the form of alanines. And I told you that yesterday in very clearly and very conceptually I told you. And along with that, we are also done with the enzymes, that is AST and ALT, sir. Okay? So once you're done with the transamination here, after that, the next thing that was left out was urea cycle, sir. So we have to now discuss about what, sir? Urea cycle, sir. So ammonia will be going to the liver, and after going to the liver, we have to learn what's the urea cycle right now. Now, the first topic to begin today will be the urea cycle, sir. And urea cycle, first learn the another name. It is also known as ornithine cycle. Urea cycle, it is also known as ornithine cycle, or else one more name of this one will be the Krebs-Hanslade cycle, sir. Urea cycle is also known as Krebs-Hanslade cycle. The urea cycle or ornithine cycle or Krebs-Hanslade cycle, sir. First, you learn the another name of this one. And location, we already know very well. Yes, urea cycle will be taking place in the liver. But remember, in the liver, it is taking place both in cytosol as well as mitochondria. So few reactions are taking place in cytosol and few reactions are taking place in mitochondria, sir. And it is a very small cycle. Actually, there are like only 5 reactions in this one. And we are going to learn all that 5 reactions right now. Is it okay? So let's get started with the reactions right now, sir. Urea cycle is going to begin with ammonium ion. Ammonium ion, that is NH4 plus. And this ammonium ion combines along with carbon dioxide, and both of them will combine together to form carb-amyl phosphate. Ammonium ion will combine along with carbon dioxide to form carb-amyl phosphate. And because the carbamoyl phosphate is forming here, the name of the enzyme here will be, of course, very simple, carbamoyl phosphate synthase 1. That is known as CPS1, sir. Carbamoyl phosphate synthase 1. आपको बस synthase लगाना है because carbamoyl phosphate is forming this, sir. Now, what is going to happen? This carbamoyl phosphate is going to join along with ornithine. So when carbamoyl phosphate combines along with ornithine, together it is going to form what's it? Citrulline. Carbamoyl phosphate combines with ornithine to form what's it? Citrulline. And the name of the enzyme here will be OTC. Now, what is this OTC here, sir? O for ornithine, T for trans, and C stands for carbamylase. Ornithine transcarbamylase. And that will lead to the formation of what's it? Citrulline. And citrulline is in turn going to combine along with aspartate. And when citrulline joins along with the aspartate, it will actually lead to the formation of arginosuccinate. Arginosuccinate. So my dear doctors, I request all of you to first learn these 3 reactions right now, sir. So reaction number 1, we are able to see there, ammonium ion combines with carbon dioxide to form carbamoyl phosphate. Carbamoyl phosphate combines with ornithine to form citrulline. That is reaction number 2. Citrulline combines along with aspartate. Together it is going to form arginosuccinate. So what I'm trying to tell you is that the reaction number 1, 2, and 3, it's all like, combining reaction. We're going to combine, and combine, sir. Now combining is done here. Let us now break down. Now what is going to happen? Arginosuccinate will actually break down to form arginine. And with the release of fumarate, Arginosuccinate will break down to form Arginine with the release of fumarate, sir. And this Arginine will again, convert back to ornithine with the release of urea. Of course, it is a urea cycle. So vapas, again it is going to form the ornithine back. And from there, obviously the another substance should be definitely urea. Why? Because in urea cycle, you can see that till now urea is not formed. So in the last reaction, definitely urea will be formed, sir. So this is your reaction number 4, and this one is the reaction number 5, sir. So as I promised you, totally how many reactions are there in the urea cycle, sir? 5 reactions are there in the urea cycle. Done, done, and done, sir. Okay. Now, out of these 5 reactions, I gave you the name of the enzyme only for the first 2 reactions only, sir. The first reaction CPS1, and the second one I gave you the name of the enzyme as OTC, ornithine transcarbamylase. Okay, now third, fourth, and fifth I did not give you the name there, sir. Why? Because you can make the name right now. Now in the third reaction, because arginosuccinate is forming here, therefore the name of the enzyme is arginosuccinate synthase. Arginosuccinate synthase, sir. And the last 2 reactions, that is fourth and fifth one, of course, we are going to break down there. And yes, Dr. Rahul, you're absolutely correct there. Because we are breaking down, we just have to add A's there. Okay, no. So reaction number 4, we are going to break down arginine. So therefore, it is arginosuccinase. That's all. And here it is arginine. So therefore, it is arginase. That's all. So fourth and fifth reaction we are going to break down. Breakdown के लिए हमको सिर्फ A's लगाना होगा सर, arginase. That's all. Is it okay? Fine here. So by this we are done with all the 5 reactions of the urea cycle. Perfectly done here, sir. आराम से हो गया. ठीक है ना? That's all. Now after completing the entire urea cycle, now my dear friends, let us discuss about the MCQs right now. Now, MCQ number 1, can anyone tell me what is the formula of urea, sir? चलिए बताइए सर, यूरिया का फॉर्मूला क्या होगा? Remember, the formula of urea will be NH2CO NH2. Yes, very good, Dr. Saurav there. The formula of urea will be NH2CO NH2. But in your exams, they're asking the question in depth. Like, not only just learning the formula, you also have to remember from where are you getting that NH2, from where are you getting the CO group, and from where are you getting like one more NH2 group, sir. Okay? So remember, my dear friends, in the very first reaction, we have added ammonium ion here, NH4. Ammonium ion. That ammonium ion is actually going through all these reactions, reaction number 1, 2, 3, 4, and 5, and finally contributing to this NH2 group, sir. Oh. So the first NH2 group is coming from ammonium ion. Is it make sense? Yes. And in the very first reaction, we are adding carbon dioxide. Carbon dioxide will also undergo all that reaction number 1, 2, 3, 4, and 5, and finally contribute the carbon CO group here, sir. Okay. Now, this is like very easy, sir. Average MBBS student will understand these 2 things. Now, the important thing is from where are you getting like one more NH2 group from you? So from where are we getting this one more NH2 group, sir? Please look at the reaction number 3. In the reaction number 3, we added the aspartate. Now, what is aspartate? It is nothing but your aspartic acid. Aspartate is nothing but what's an aspartic acid? It's an amino acid. Yesterday we have seen in the classification, it's an acidic amino acid. Yes, very good. Very nice. And the moment I'm telling you amino acid, automatically you should get already in your mind that amino acid means what is the structure of that one? Carbon in the center with all that 4 bonds there, sir. And on one of the bond, there will be amino group attached. So if you're adding the aspartic acid, it means that indirectly you're adding what there, sir? Amino group. So if you're adding the aspartic acid there, amino group आप add कर रहे हो. ठीक है? Done here. So therefore, my dear friends, now write down below here, what will be the sources of urea? The formula of urea will be NH2CO NH2. And in this, the first NH2 group is obtained from ammonium ion. The CO group is obtained from carbon dioxide. One more NH2 group is obtained from aspartate. That's very, very important concept. The formula of urea is NH2CO NH2, and then NH2 group is obtained from ammonium ion. CO group is obtained from carbon dioxide. And one more NH2 group is coming from where, sir? It's coming from aspartate. Perfectly done. Hello, sir. Are you done? Are you all done, my dear friends? Now please come back to the very beginning here. In the beginning of this urea cycle, I told you very, very clearly the urea cycle is taking place in the liver. And in the liver, few reactions will be taking place in cytosol, few in mitochondria. So now we have to learn like which reactions are taking place in the cytosol and which reactions will take place in mitochondria. And for that, it's very, very easy. Now in this diagram, what you do is please draw a line from here like this. S. Understanding? It means that the first two reactions will be taking place in mitochondria and the remaining three reactions will be taking place in cytosol. The first two reactions will be taking place, that is CPS1 and OTC, will be taking place in mitochondria, whereas the remaining three reactions will take place in the cytosol, cytoplasm. So, koi dikkat? Come on, sir. Koi dikkat? So in this manner, we are covering like all the points regarding the urea cycle, sir. And one last and final MCQ which is remaining here is, yes, my dear friends, where are you going to control this urea cycle? आप इसको control कहाँ पर करेंगे? Rate limiting enzyme. And the rate limiting enzyme of urea cycle will be CPS1. So CPS1 will be the rate limiting enzyme of the urea cycle. Perfectly done, sir. Okay. So the rate limiting enzyme of the urea cycle will be CPS1, sir. Badhiya, sir. And by this, we have covered all the points regarding the urea cycle. And you know very well, urea will be actually excreted out in the urine, sir. Fine. That's perfect. Yes, Dr. Shinit, it is actually PFK1. PFK1, Dr. Shinit. Okay? That's all. So the overview that we actually started yesterday, I think we have completed that placing. And overview यहाँ पर ये complete हो चुका है. The first two reactions will take place in mitochondria. See, we have drawn the diagram right in the lines there. Outer membrane will be smooth and the inner membrane will be having folds known as cristae. So from that diagram, from that line, you can actually understand that the first two reactions are taking place in mitochondria. So reaction number 1 and 2 will be in mitochondria and 3, 4 and 5 will be in the cytoplasm. If you want, I'll just mention that here also. So in mitochondria, reaction number 1 and reaction number 2. And in cytoplasm, there will be 3, 4 and 5. Clear, guys? Sure. And by this, we are done with what, sir? Transamination. Yesterday, we have completed. Like how is the ammonia transported? And today, we have actually completed what here, sir? About the urea cycle. Urea cycle is also done here, sir. ठीक है ना? चलिए, अब हम बढ़ते हैं आगे. Let's start with the new topic right now, sir. That is what we were discussing since yesterday. The overview of the protein metabolism done. Now, please write down there the heading as metabolism of aromatic amino acids. Can you please recall from our yesterday's classification? What are the aromatic amino acids, sir? Aromatic amino acids, there will be 3 of them. Number 1, there will be tyrosine. Number 2, there will be tryptophan. And number 3, there will be phenylalanine. Tyrosine, tryptophan, and the phenylalanine. These 3 will be the aromatic amino acids. Done. Now, how we are going to approach this topic is, first of all, we are going to learn about the tryptophan, sir. So first, we are going to learn separately about the tryptophan, and then we are going to learn about the phenylalanine and tyrosine separately. So that metabolism is separate. So I request all of you to please write down there tryptophan. Now, tryptophan is going to form 3 important substances. Number 1, Tryptophan is the one which is going to form melatonin. Number two, tryptophan is the one which is going to form serotonin. And I hope you all know very well, serotonin is also known as 5-HT, 5-hydroxytryptamine. Many questions come from this, sir. Yes, of course, Dr. Saurabh. Yes. So tryptophan is the one which is going to form melatonin and serotonin, sir. So please remember with the mnemonic like this, tryptophan is going to form melatonin and serotonin. So please remember, tryptophan is also going to form melatonin and serotonin. And the third one is very much important here, sir. Tryptophan is the one which is going to form your vitamin B3, which is also known as niacin, or else which is also known as nicotinic acid. Very, very important. It's an amino acid. Tryptophan is an amino acid, and an amino acid is going to form a vitamin. That is how the question was asked in the exam, sir. Amino acid forming a vitamin. Which amino acid is going to form the vitamin? Tryptophan, sir. Tryptophan is the one which is going to form vitamin B3. And one more thing you mentioned here itself, my dear friends, remember 60 milligrams, 60 mg of tryptophan is going to form 1 milligram of niacin. That is your vitamin B3. Even that is also asked in your exam, sir. 60 mg of tryptophan is going to form 1 milligram of niacin, sir. Done. That's all. So you just have to remember the three important products obtained from here, sir. Now, once you are done with this one, let us write down the clinical correlation. Now, here there is a disease. The name of the disease is heart nerves disease, sir. अब यार ये disease में होता क्या है? Remember, in heart nerves disease, there will be decreased absorption of amino acid from intestine. Normally, what is going to happen after eating the food? Once you consume the food will be digested. And suppose you are going to consume the protein-rich diet. So yesterday you told me about that chicken, mutton, soya and all. So proteins are getting digested to amino acid. And what is the next thing that should happen? From the intestine, that amino acid has to be absorbed. But in this, amino acid will not be absorbed, sir, in the heart nerve disease. Okay, no? So there will be decreased absorption of amino acid from the intestine. And mainly, which amino acid will be decreased, sir, in this disease? That is, tryptophan will be decreased. So there will be decreased tryptophan. In heart nerve disease, there will be decreased amount of tryptophan. अब आप इसको ना ऐसे समझो. Now try to understand here like this, sir. Please look at the screen, everyone. For example, in heart nerve disease, if tryptophan is reduced in our body, that means it is unable to form what, sir? Vitamin B3. And if vitamin B3 will not be there, yes, you know very well, deficiency of vitamin B3 will lead to pellagra. Pellagra, the very famous one. है ना? तो यहाँ पर भी same वही होगा ना? So what is the clinical feature of the heart nerve disease? Remember, in heart nerve disease, you'll be having pellagra-like symptoms. Same pellagra-like symptoms will occur in the heart nerve disease, sir. And I'll tell you about that pellagra in detail in the vitamins topic, sir. In vitamins topic, I'll teach you about that one, sir. Yes, that 3D disease, dementia, diarrhea, and all those things, a very important one. I'll teach you in that one, sir. Taken? So that is all that you have to remember about the tryptophan, sir. Tryptophan topic completed here. Now, once you are done with the tryptophan here, what is the next thing which is left out, sir? The tyrosine and the phenylalanine. Now starts a bigger topic, sir. So please write down the heading there as metabolism of phenylalanine and tyrosine. Metabolism of phenylalanine tyrosine. Now, I told you very clearly, this is actually a bigger topic here, sir. So have some patience. First of all, we are going to see the overviews here, sir. Let's see the overview कि हमको यहाँ क्या-क्या पढ़ना होगा. So, my dear friends, first of all, there will be phenylalanine. Phenylalanine is obviously an essential amino acid. You know, na essential amino acid. TV till 8 p.m. and P stands for phenylalanine. And essential means what, sir? Are they produced in our body? No, they are not produced in our body. They have to essentially be taken in the diet. Okay. Now, once you take that phenylalanine in the diet, what is going to happen? Phenylalanine will now get converted to tyrosine. How? By addition of OH groups. And yesterday you people only told me, right? What is the name of the OH group? OH group stands for hydroxyl group. Hydroxyl group. So from there itself, you can easily remember the name of the enzyme. If you're adding the hydroxyl group, the name of the enzyme will be hydroxylase. And how to make the full name? Add the name of the substrate. साथ में substrate का नाम लगा दो, phenyl alanine hydroxylase, PAH. So phenylalanine is getting converted towards a tyrosine by addition of OH group. And OH group is nothing but what's a hydroxyl group. That is why what is the name of this one? That is phenylalanine hydroxylase. Phenylalanine hydroxylase. And now once this tyrosine is formed, tyrosine is very much important one. Why? Because it is going to form, number one, melanin. See, that is the reason why I gave you that mnemonic. Don't get confused from today onwards. Tryptophan is the one which is going to form melatonin. Tryptophan is going to form melatonin. Whereas tyrosine is the one which is going to form melanin. And melanin is the pigment present in our skin, in the eyes, in the hairs, and all. It is a pigment. Whereas melatonin is the hormone which is released from the pineal gland. And that is going to maintain your circadian rhythm, the sleep-wake cycle. So melatonin is different, melanin is different. Okay, na? Number two, tyrosine is the one which is also going to form the thyroid hormone T3 and T4. And number three, tyrosine is also going to form catecholamines. And what are catecholamines, sir? Norepinephrine, epinephrine, as well as the dopamine, sir. Dopamine, norepinephrine, and epinephrine. These three are together known as catecholamines, and they are also formed from tyrosine, sir. So are you able to see how many important substances are being formed from here, sir? देख रहे हो आप? So now please put this in a box here like this, sir. This is just the overview. And now in this overview, we have to learn totally like 4 topics here right now, sir. Topic number 1, we are going to learn about the phenylalanine to tyrosine. And then topic number 2, tyrosine to melanin. Then tyrosine to T3, T4, and then tyrosine to catecholamines, sir. So we are going to learn all these 4 topics right now, sir, one by one. Yes, doctor, DA stands for dopamine there, dopamine. Norepinephrine, epinephrine, and dopamine. Okay, na? So chaliye, now one by one, let's start working out on all the 4 topics, sir. The first topic, very easy one. Conversion of phenylalanine to tyrosine, sir. And you already know very well, phenylalanine is getting converted to tyrosine in the presence of an enzyme known as phenylalanine hydroxylase. Achha. Addition of OH group. But you have to remember here, okay, who is adding that OH group, sir? Remember, the OH group will be added by tetrahydrobiopterin. And after adding the OH group, it is going to become dihydrobiopterin. So we can also write it like this here, tetrahydro, and that is going to become what's a dihydro. Are you getting the clarity now? So phenylalanine is getting converted to tyrosine by addition of OH group, sir. By addition of OH group. And OH group will be added by tetrahydrobiopterin, and that is going to become dihydrobiopterin. That is only one new thing that you have to learn here, sir. That's all. Remaining everything to aapko already pata hai. Now, what is going to happen now? Let us see the clinical correlation. In some unfortunate people, in some unfortunate babies, what is going to happen, sir? Biopterin, RR. Dr. Vishal, it is biopterin. Okay. Now, Now, in some people, what is going to happen? Phenylalanine hydroxylase enzyme is deficient. If this enzyme is deficient, common sense, this reaction will not occur. Common sense, phenylalanine level will go up in the blood and that will start excreting in the urine also. And that condition is known as phenylketonuria. One of the important ones, sir. So clinical correlation. Phenylketonuria. Phenylketonuria, that is PKU. Okay. Now, in phenylketonuria or any disease, whatever disease we're going to learn, sir, first you write down there as enzyme deficient. Now, what is the enzyme deficient in phenylketonuria? Phenylalanine hydroxylase. The enzyme deficient in phenylketonuria will be phenylalanine hydroxylase. Okay, phenylalanine hydroxylase. Okay. Now, after that, what are the clinical features of phenylketonuria, sir? What will be the clinical features of phenylketonuria? Let us see this one by one right now, sir. And the best way to learn this is with the help of my overview here, sir. So let me repeat that once more here, sir. Phenylalanine hydroxylase is deficient. If this is deficient, then this reaction will not occur. Then phenylalanine levels will go up in the blood and that will also get started excreting in the urine. And that disease is known as what, sir? Phenylketonuria. Fine. Done. But we have actually seen all these things in the reverse manner, so retrograde manner. I mean to say before here. Now let us see further. Aage kya hoga? Now, my dear friends, remember, if this reaction doesn't occur, obviously tyrosine form nahi hoga. If tyrosine is not formed, obviously melanin will not be formed, sir. यहाँ पर melanin form nahi hoga, sir. Are you understanding the importance of my overview here? If tyrosine is not formed, then melanin will not be formed. Now, you people tell me one thing here. If melanin is not formed, sir, we all know melanin will be present in, first of all, it is a pigment. Okay, no? It is present in what, sir? It is present in the skin. It is a pigment present in skin. Now, you all know very well if the melanin increases in the skin, that will be leading to hyperpigmentation. And if melanin decreases in the, skin, it will lead to hypopigmentation. It is leading to hypopigmentation. Okay, na? Dr. Stuti, please don't say albinism. It is not albinism. Dr. Najib Ansari, please don't say albinism. Albinism, Vandana, please don't say albinism. Albinism and vitiligo is a different thing. It will occur due to deficiency of a different enzyme. I'll tell you within few minutes. Don't worry about that. So here you don't say albinism. If melanin increases, hyperpigmentation. If melanin decreases, hypopigmentation. So therefore, what is going to happen here? Hypopigmentation. Okay, so therefore, the patient is going to first come to you with pale skin. The skin will be very much pale. Pale skin, sir. Okay, then after that, my dear friends, remember, melanin is not only present in the skin, melanin is also present in the hairs also. और अगर hairs में melanin का कमी हो जाएगा, if there is a decreased melanin in the hairs, then what is going to happen? That is going to become like blonde hairs, sir. Don't say white hairs, blonde hairs. और blonde hairs का मतलब क्या होता है? That will be, yes, golden yellow color. Golden yellow color हो जाते हैं hairs, है ना? Blonde hairs. So the best way to remember this one is like, remember one, Russian in your mind, sir. You're all like foreign medical graduates. You have seen Russians a lot. Forget about seeing. You have even like, खैर आपका अनुभव कुछ ज्यादा है तो आप सबने फील भी किया है ना? हाँ, तो आपने रशियंस को देखा होगा. So they'll be having like pale skin. The skin will be very pale, है ना? And then the hairs will be golden yellow color. Blonde hairs होते हैं. Acha, coming back to the overview here. If tryptophan is not formed here, remember, not only melanin, even T3, T4 will also not be formed, sir. T3, T4 will also be not formed, sir. और अगर T3 T4 फॉर्म नहीं होता है तो रिमेंबर thyroid hormones they are actually necessary for the proper development of your nervous system, sir. Thyroid hormones are necessary for the proper development of your brain. And if they are not there, the brain will not develop properly. So therefore, what is going to happen? It will lead to mental retardation. Oh my God. Are you understanding here? Mental retardation hota hai, sir. So what is going to happen here, sir? Initially, when you learn the first two symptoms, the disease looks very good. Oh, naturally we are getting like pale skin. Naturally, we are getting blonde hairs. We don't have to spend a lot of money in the, beauty parlors and all this. But, my dear friends, don't be happy that disease is so fascinating, very nice one. Even though you're having pale skin, even though you're having blonde hair, remember mental retardation, sir. So you'll be like mentally retarded. ये कुछ काम का नहीं होता है. Are you understanding the point here? Next one, after that, the next thing here will be, yes, there will be even a characteristic odor also, sir. And the characteristic odor from these people will be like mousy odor or musty odor in the urine. Mousy or musty odor in the urine, sir. Mousy or musty odor. Mousy, the word is coming from mice. Rodents, rats, है ना, से गंदी सी स्मेल आती है. तो इसीलिए आई एम टेलिंग यू टू रिमेंबर द एग्जांपल एज अ रशियन हियर, सर. आप ये सारे पॉइंट याद कर सकते हो बड़ी आसानी से. विल बी इजीली एबल टू रिमेंबर. तो फिनाइल कीटोन्यूरिया आज से याद रख लो. रशियन, पेल स्किन, ब्लॉन्ड हेयर. दिमाग से तो वैसे ही पैदल होते हैं वो लोग, है ना. तो मेंटल रिटार्डेशन. एंड देन माय डियर फ्रेंड्स रिमेंबर there'll be bad smell, that is musty or mousy odor from here, sir. Yes, of course, Dr. Shubham. Are you all done? चलिए सर, बढ़िया. And this is what you have to remember about the phenylketonuria, sir. चलिए, हो गया. I hope you all remember my overview very well here, sir. In this overview, we are done with the first topic, conversion of phenylalanine to tyrosine, sir. ठीक है ना? Conversion of phenylalanine to tyrosine. So therefore, this topic is done here, sir. Now, welcome to the second topic here, tyrosine to melanin, sir. Tyrosine to melanin, guys. ठीक है ना? Now, suppose these are the characteristic features. If you remember this one, that is more than enough, sir. Okay, na? Nothing else about that one. Dr. Abhilash Reddy is telling, sir, again you are saying about Russians. You mean I should not say? Sir, can you kindly tell where in intestine most of the tryptophan is absorbed? Jan FMG, I wrote jejunum. Yeah, normally tryptophan is absorbed from the small intestine. ऐसा कुछ specific नहीं है. Specifically, we'll be learning about the absorption of iron, absorption of vitamin B12 from the terminal part of the ileum, and folic acid. फोलिक एसिड के अब्जॉर्प्शन के बारे में हम पढ़ते हैं जेजुनम से. फोलिक एसिड होता है जेजुनम से. सो नॉर्मली अमाइनो एसिड्स आर अब्सॉर्ब फ्रॉम द इंटेस्टाइन एपिथेलियल सेल्स, एंटेरोसाइट्स. चलो सर, हो गया, डन. नेक्स्ट वन सर. Now, my dear doctors, we are done with the first topic here. Now, welcome to the second topic right now, sir. ठीक है ना? That is the formation of melanin. अब पढ़ते हैं formation of melanin, sir. So please write down the heading there as formation of melanin, sir. So melanin is formed from which amino acid? Tyrosine. Tyrosine is the one which is going to form melanin. And this is so easy. The name of the enzyme here will be tyrosinase. Wow. Tyrosine is going to form melanin in the presence of an enzyme known as tyrosinase. Just you have to add the A's there. Now, again, the same thing. In some people, unfortunately, this enzyme will be deficient. This reaction will not occur. Melanin will not at all be formed, sir. Guys, see, there are like two things here. Decrease melanin and deficient melanin. अगर melanin का कमी होगा, if melanin is decreased, then it will lead to hypopigmentation, that is pale skin. अगर melanin form ही नहीं होगा, if melanin is not at all formed only, then forget about hyper or hypopigmentation, pigmentation ही नहीं होगा. Pigmentation will not occur. So therefore, that will be having like white patches on the skin, and that condition is known as albinism. Understanding? So in albinism, which enzyme is defective? The enzyme defective will be tyrosinase. It comes here, sir. Understanding? So please write down there as clinical correlation. And here the clinical correlation will be albinism and even the vitiligo, sir. So this is due to deficiency of which enzyme? Enzyme deficient will be tyrosinase. Thank you, Dr. Stuti. So if tyrosinase enzyme is deficient, if it is defective, it will lead to albinism. So by this topic is also done. Now again, welcome back to this overview here, sir. In our overview, we are now done with what's the formation of melanin. This topic is also done. And now let's go to the next topic, that is formation of T3, T4. So the next topic that we are learning is formation of T3 and T4. Thyroid hormones. And this topic, my dear friends, it is very, very important topic. You know the importance of thyroid hormones. है ना? Thyroid hormones का importance आपको already पता है. And it's very, very common. Like when you start practicing, yes, when you start practicing, my dear friends, these patients are going to very commonly come to you. And especially in the females, hypothyroidism is very common. है ना? ये बहुत common है. तो इसीलिए, my dear doctors, remember, thyroid hormones metabolism, we have to know in detail as a doctor. As a doctor, we have to know it like very, very clearly. Very clearly. Understanding? So let us learn about the thyroid hormones in detail, in detail from the scratch. Okay. चलिए start करते हैं sir. Now, first of all, imagine this one to be your thyroid gland here. And we know that thyroid gland is going to secrete three hormones, T3, T4, and even calcitonin. Thyroid hormones are going to secrete three of them, that is T3, T4, and calcitonin. Is it okay? These are the three hormones secreted by thyroid gland. And now let us go deep inside the thyroid gland, like microscopic structure. Let us see the histology of that microscopic structure. So inside the thyroid gland, you'll be actually having round follicles here. And these follicles will be lined by cells. And these cells are known as the follicular cells. So the follicle will be lined by the follicular cells. And then in the middle of this follicle, you'll be actually having the cavity, and that cavity is known as the follicular cavities. So this one here will be the follicular cells. And in the center, there will be a cavity, and this cavity is known as what's a follicular cavity. Perfect. Then apart from that, you will also have certain cells which are present around the follicle cell. There are certain cells which are present around the follicle. And these cells which are present around the follicles here, these are known as the parafollicular cells. And these parafollicular cells, they're also known as C cells. The parafollicular cells, they're also known as C cells. So पहले आप ना आराम से, neat and clearly, you draw this diagram and write down the notes there. So basically, what are you understanding here, sir? That inside the thyroid gland, you'll be actually having two types of cells. That will be the follicular cells and the parafollicular cells, sir. Inside the thyroid gland, there'll be two types of cells. That is your follicular cells and the parafollicular cells, guys. Now, out of these two types of the cells here, remember, follicular cells are the one which are going to form T3 and T4. Whereas parafollicular cells are the one which are going to form calcitonin. Oh my God, are you understanding? So as a medical student, you have to know this anytime, every time by default. By default, आपको ये पता होना चाहिए sir. Is it okay? So follicular cells are the one which are going to form the T3 and T4, and the parafollicular cells are the one which are going to form calcitonin. This you should know by default, sir. So यार अब बताओ what is our topic right now here? Our topic is about the formation of T3, T4, and T3 and T4 is forming from where? From the follicular cells. So therefore now let us zoom the follicular cells, sir. Let's see inside the follicular cell. Follicular cell के अंदर देखते हैं, ठीक है ना? तो चलिए फॉलिकल को हम जूम करते हैं और फॉलिकल के अंदर देखते हैं. So let's see inside the follicle. So now, my dear doctors, imagine this is like a big follicle here. The follicle is lined by follicular cells here. Okay. So let me write down the name again here, sir. This one here will be the follicular cell. And in the center, there will be a cavity, and that cavity will be the follicular cavities. And near to this follicle, you'll be actually having a blood vessel. First, you draw the diagram aram se. Take your time. So there is a follicle. Follicle will be lined by follicular cells. In the center, there will be a cavity, follicular cavity. And of course, nearby, there will be a blood vessel. You just imagine any capillary present over this. My dear friends, confirm me if you are ready with the diagram. Then I'll start the kahani. Done? Ready? Thank you. Very nice. It's like a nice story, sir. So you just remember like a story here. It's okay now. So inside the follicular cell, there will be formation of a protein known as thyroglobulin. Inside the follicular cell, there will be formation of a protein known as thyroglobulin. After the formation of this protein thyroglobulin, it will be transported out into the follicular cavity cell. Thyroglobulin will be transported out into the follicular cavity. And from that time, I'm telling you that thyroglobulin is actually a protein. If it is a protein, it is made up of amino acid, many amino acid. Somewhere in the middle, you are having an amino acid known as tyrosine. Here comes the hero of the story. Is it okay? Follicular cells, function number one, they are going to form a protein known as thyroglobulin. And after the formation of thyroglobulin, it will be transported into the cavity. And in the cavity, yes, it's a protein. It is made up of amino acid. Somewhere in the middle, there is tyrosine. You just remember, okay, acha. Among this amino acid, there will be tyrosine. Okay. Chalo. Now leave this here only, sir. Isko aise hi rehne do. Let it be there. Now, my dear friends, welcome to the next function, that is handling the iodide ions. In the blood vessels or in the blood, you'll be having iodide ion. In the blood, you'll be having iodide ions. Now, don't ask me where is this iodide ion coming from. We all know very well, we are consuming food. In the food, we are using salt, and that salt will be actually iodized salt. Hota hai. So that iodide ions will be actually absorbed into the follicular cavity. Now, how are these iodide ions absorbed into the follicular cavity? Yes, with the help of a transporter here. And that transporter is going to transport iodide ion along with sodium. Iodide ion along with the sodium. थोड़ा हम ना आराम से पढ़ते हैं सर. It's like a very good, beautiful mechanism. And why I'm telling you like slowly is why, because आप सबको पता है about the thyroid disorders, they are going to come along with you till Intel medicine, so they're not going to leave you. So it comes in physiology, then pharma, then in medicine. So everywhere it is going to come. तो इसीलिए हम लोग इसको थोड़ा सा ना आराम से अच्छे से अभी पढ़ते हैं. Let us learn it right now perfectly so that you can apply in other subjects. Now, what is the name of this transporter that I have drawn here in black color? This transporter will be known as the sodium iodide symporter. I hope all of you know that. Hello, Ji. All of you know that, my dear friends, okay, whenever any two ions are going to move in the same direction, that is known as symport. So first of all, iodide ion will be trapped inside with the help of sodium iodide symporter. Okay, now symporter. Now, whenever any two ions are going to move in the same direction, that is known as symport, and when they're going to move in the opposite direction, that is known as antiport. So the name is very simple here. Then next one. After that, this iodide ion will be taken up by the follicular cells, and then from the follicular cells, they are going into the follicular cavity now. Okay, they are going to the follicular cavity now. And here they are going with the help of another transporter now. Now, what is the speciality about this one is, it is going to transport the iodide ion. It will transport the iodide ion, but in exchange of what, sir? In exchange of chloride. So therefore, let me write down this one here very clearly. The blue colored one that I wrote there. This transporter will be iodide chloride exchanger. Iodide chloride exchanger. Yes, Najib Ansari. Yes, pendrin. आपने physiology में पढ़ा होगा. Yes, yes, of course, Dr. Shinit. So iodide chloride exchanger. अब क्या होगा? Now what is going to happen? Are you understanding beautiful thing here? So first we have taken the iodide ion from the blood vessel, from the blood into the follicular cell, and from the cell into the follicular cavity. And this is known as the trapping of iodide. We have actually trapped the iodide cell. Trapping is done. After trapping the iodide, now we are going to convert this iodide to iodine, I-1 to I. और ये reaction तो मैंने आपको कल बताया था. Yesterday only I taught you this one in the very beginning of biochemistry. I told you iodide to iodine will be an oxidation reaction. And if it is oxidation reaction, of course, it has to be done with the help of an enzyme. And that enzyme will be TPO. Now, what is the full form of this TPO here? It is thyroperoxidase. Okay. So iodide to iodine, the name of the enzyme will be TPO, and TPO stands for thyroperoxidase. Thyroperoxidase. Okay. So iodide ion is first of all going to come into the cell, and from the cell it is going to come into the cavity, and then iodide to iodine, it is oxidation done, sir. Chali, badhiya. Now you tell me one thing. What do you have in the follicular cavity right now? You have iodine in the follicular cavity right now. And not only iodine in the follicular cavity, you also have tyrosine in the follicular cavity, which is already formed. It is already present there. Where do you have all these things right now? So follicular cavity. That's all. So what you do is you actually make the concept like very clear in your mind. Just I taught you about two things, sir. First, we are going to bring in thyroglobulin, and then we are going to bring in iodine. So we are going to bring thyroglobulin and we are going to bring what's up? Iodine. Ab kya kiya jaye? The third and the final step, the last step, join them together. So let us join them together right now, sir. So this iodine will now go and get attached towards the tyrosine. ये iodine किसको attach होता है? Tyrosine को attach होता है. कैसे attach होता है? How it is going to attach? Sometimes one iodine will be attached to tyrosine. So it is known as mono iodo tyrosine. Very simple. MIT. Sometimes two iodine will join with tyrosine. So therefore it is डाई आयोडो टायरोसिन, और इसको हम बड़े प्यार से DIT कहते हैं. So one will be MIT, another one will be DIT, monoiodotyrosine, and then diiodotyrosine. And then what is going to happen? One molecule of MIT will be joining with one molecule of DIT. So MIT plus DIT. Tell me how many iodines now? 3. So if there are 3 iodines here, so therefore it will be triiodo Tyronin. Be careful with the name. The name changes here. It is no more tyrosin now. It is not tyrosin. It is thyronin. Triiodothyronin. और इसी को हम बड़े प्यार से T3 कहते हैं. So you have nicely mugged up as T3, T4, T3, T4 from your school days. Please learn the full form also now, sir. Achha. Now, sometimes what is going to happen, one molecule of DIT and one more molecule, 2 molecules of DIT will combine, sir. So DIT plus DIT. So in total, how many iodines right now, sir? Tetra. Tetra iodothyronin. And this tetraiodothyronin is nothing but your T4. इसी को हम T4 कहते हैं. And that itself is known as what, sir? Thyroxin, sir. Tetraiodothyronin itself is known as what, sir? Thyroxin. That itself will be thyroxin, sir. Are you done? चलिए बढ़िया, sir. So in this manner, we are done with the formation of T3, T4. तो आपको क्या करना होगा? What you have to do here right now, sir? It's very simple. It's like 1, 2, 3. Number 1, we are going to bring the thyroglobulin into the cavity. Number 2, we are going to trap the iodine and oxidize the iodide ion to iodine. So number 1, bring thyroglobulin. Number 2, bring iodine. और उसके बाद क्या किया जाए? Time pass. Join them together. It's all conjugation reactions. Okay, na? Thank you, Dr. Umesh. Thank you. So simple, so simple. It's like time pass. So T3 will be triiodothyronin and T4 will be tetraiodothyronin. And this T3 and T4 are formed from which amino acid? They are formed from tyrosine amino acids. Crystal clear concept. Now, once you are done with this one here, I hope, my dear friends, you all remember my overview still? याद है कि भूल गए? We are actually working out on which overview? TPO stands for thyroperoxidase. Follicular cell and the follicular cavity के बीच में ये जो blue color वाला मैंने dot जो draw किया है, that is your iodide chloride exchanger. And this transporter, that black colored one, is sodium iodide symporter. And this orange color one जो हमने लिखा है, this is the enzyme TPO. TPO stands for thyroperoxidase. Do you all remember this overview, sir? Hello, G. So now we are actually done with the melanin. And after completing about the formation of melanin, now T3, T4 is also done. And the only thing that is left out right now, my dear friends, is catecholamines. और ये तो बड़ा आसान है, sir. It's very, very simple. चलिए start करते हैं, sir. Formation of catecholamines. Formation of catecholamines. And this topic is like so easy, so easy. It's like time pass. Yeah, of course, Dr. Rekha. That is nothing but in the follicular cavity only. That is your thyroglobulin only. Yeah, that protein. First of all, you're having phenylalanine. और अभी अभी हमने पढ़ा था, just now we have learned phenylalanine is going to form tyrosin. How? By addition of OH group. That is the first reaction we have learned in the very beginning of this topic. And the name of the enzyme will be phenylalanine hydroxylase. Acha. because we are adding OH group, hydroxyl group, that is phenylalanine hydroxylase. Today we completed biochem. No, I think today will not be able to complete the entire biochem. I think 2-3 hours will be left. एक 2-3 घंटे का बच जाएगा, and that will be doing it tomorrow. Tomorrow, do you have any class? You don't have any class. So एक 2-3 घंटे हम कल कर लेंगे क्लास आराम से, ठीक है ना? Yes. And I'll be actually even traveling back to Hyderabad tomorrow. I'm not at Hyderabad here. Actually, I'm at other place here. I'm at a medical college here. तो morning में actually ना थोड़ा recording हो रहा है. Even I'll be back to Hyderabad tomorrow morning flight है. So आराम से तब हम कर लेंगे वहां से, ठीक है ना? चलो नेक्स्ट वन सर. तो phenylalanine is getting converted to tyrosine. ये हो गया. Now tyrosine इसमें क्या होगा? We are going to add one more OH group, sir. So when you add the OH group, this is going to become what, sir? DOPA. Now you tell me what should be the name of the enzyme. Again, the same thing. We are going to add the OH group, hydroxyl group. Okay, no? So because we are adding the OH group, the name of the enzyme will be hydroxylase only. And the complete name of the enzyme will be tyrosine hydroxylase. साथ में substrate का नाम लगा दो. Tyrosine hydroxylase. ठीक है, done. And finally, there is a formation of DOPA. DOPA form हो रहा है. Now, can anyone tell me what is the full form of DOPA here? Shubham Thakur is writing business class. अरे यार डोमेस्टिक में कहाँ से बिजनेस क्लास पे? गरीब आदमी. बाई रोड जा रहा हूँ मैं, बाई रोड टैक्सी लेके. हाँ. So now what is the full form of DOPA here? See, what we have done is initially we have taken phenylalanine. In phenylalanine एक बारी मैंने OH group ऐड किया और 2 बारी मैंने OH group ऐड किया दूसरी बार. So 2 times I have added the OH group. 2 times we have added the OH group. So therefore it is known as what? इसलिए इसको क्या कहा जाता है? Dihydroxy. Because 2 times you have added the hydroxyl group. Dihydroxy phenylalanine. Dihydroxy phenylalanine. हो रहा है डाइहाइड्रोक्सी फिनाइललाइन, ठीक है ना? इट्स वेरी सिंपल. ये तो बड़ा आसान है. नाउ आफ्टर दैट फ्रॉम दिस डोपा यू आर गोइंग टू रिमूव द कार्बन इन द फॉर्म ऑफ कार्बन डाइऑक्साइड. So what is the name of the enzyme? Removing D, carbon carboxylase. So it is decarboxylase. And the full name will be DOPA decarboxylase. So from the DOPA, we are going to remove the carbon dioxide, DOPA decarboxylase. And that is going to now become what, sir? Dopamine. मेरी नजर से देखो. We got the first catecholamine. डोपामिन मिल चुका है, इसलिए मैं आपको बार बार कह रहा हूं ना. तो that is the reason why I'm telling you repeatedly here. This topic is like very, very easy, sir. Next. In this dopamine, again, you are going to add one more OH group that is going to become norepinephrine. Is it okay? So if you are going to add once more OH group, it is going to become norepinephrine. And OH group, my student knows very well about the OH group, sir. Hydroxyl group. इसीलिए एंजाइम का नाम क्या होगा? डोपामीन हाइड्रोक्सिलेज. और मेरी नजर से देखो दूसरा कैटेकोल अमाइन भी मिल चुका है. We got the second catechol amine. Lovely. And now, my dear friends, if you observe this entire pathway is so simple here. The entire pathway is like so easy here. Everywhere it is like we are adding OH, OH, and OH groups. Hydroxyl, hydroxyl, and hydroxyl group only, except for one place, that is DOPA. And DOPA, we are going to remove the carbon. और उसको आप आराम से याद रख लेना. Remember as DD, DOPA decarboxylase. DD, DOPA decarboxylase. ठीक है ना? Then after that, my dear friends, next one more thing here is that, final thing here is that, norepinephrine will be actually getting converted towards epinephrine. Norepinephrine will be getting converted to epinephrine, sir. Now, how is this norepinephrine getting converted to epinephrine? How it is getting converted, sir? This reaction is extremely important. I'm teaching you for this reaction only, sir. This entire pathway, think like I'm teaching you for this reaction only. So remember, it is by methylation. Which reaction, sir? Methylation reaction. So addition of methyl group, sir. Methyl group यहाँ पर add होता है. But now the most important thing is who is going to add that methyl group. So remember Sam. Sam will add the methyl group. अब यार ये Sam कौन है? Sam is S-adenosyl methionine. Who is Sam? Sam is S-adenosyl methionine. It is going to add the methyl group. ये methyl group add करेगा. So it is going to add the methyl group here, sir. CH3. Now Sam will add the methyl group. After adding the methyl group there, sir. After adding the methyl group, what is going to happen? The SAM will actually get converted to SAH. SAH. And that SAH stands for S-adenosyl homocysteine. So SAM will add the methyl group. And after addition of methyl group, the SAM itself will get converted to what, sir? S-adenosyl homocysteine. S-adenosyl homocysteine. तो SAM यहाँ पर आता है, अपना methyl group दे देता है. और इसको याद रखने का तरीका ये होता है कि normally I tell in my offline classes, the students remember like this here, sir, कि SAM यहाँ पर आता है और वो अपना दे देता है. और देने के बाद में क्या होता है? Anyone is going to become homo only. So it is going to become homo, sir. ठीक है ना? I mean to say homocysteine. Yeah, of course, Dr. Shinil. All this is taking place in the adrenal medulla. Adrenal medulla में हो रहा है, as well as in the neurons also, no? Sympathetic neurons, norepinephrine, neurotransmitter and all. ठीक है ना? अच्छा, because methyl group is getting transferred here, what is the name of the enzyme here, sir? The name of the enzyme is methyl transferase. Methyl transferase कहते हैं. That's all. And आपके MCI एग्जाम में ये पूछा गया था. In your FMG exam, they have asked, who is the methyl group donor? Who will give the donor? Who will give the methyl group, sir? The methyl group donor will be SAM. S-adenosyl methionine. SAM will be the methyl group donors. Done, done, and done. And by this, we got all the three catecholamines. Done, done, done. So formation of catecholamines completed here, sir. Are you getting me here? Badhiya. And now, my dear friends, once you are done with this one here, I hope you're able to understand. Now, we are done with the entire metabolism of phenylalanine and tyrosine, sir. So we are done with the formation of tyrosine. We are done with the formation of melanin. Then we are done with the formation of T3, T4 as well as catecholamines, sir. ठीक है ना? ये सब हो गया. Now the last and final one that is remaining right now is degradation of tyrosine, sir. So please write on the heading there as degradation of tyrosine. Degradation of tyrosine. So remember, tyrosine will actually undergo degradation to form a substance known as homogentisate. That homogentisate is going to form MALEYL, mallyl acetoacetate. That is going to form fumaryl acetoacetate, and that is actually gonna break down, and that is going to break down to form fumarate and then acetoacetates. So tyrosine will actually undergo certain reactions to break down to form homogentisate. Homogentisate is going to form mallyl acetoacetate and then fumaryl acetoacetate, as well as it is going to form the fumarate and acetoacetates. Acetoacetate. Done? Are you all done? Now you might be thinking, why is sir writing continuously like this here without explaining any reactions? My dear friends, take another color pen or pencil and write down there. No need to learn this pathway. Another color pen लो और वहां लिखो no need to learn this pathway. ये pathway पढ़ने का जरूरत नहीं है. ई pathway చదవాల్సిన అవసరం లేదు. Okay. Then you might be thinking, why the hell are you teaching then, sir? So I'm teaching you only and only for one enzyme here that was asked in last year exam 2023, oxidase. And what is the complete name here? It is homogentisate oxidase. Homogentisate oxidase. Now, why is this very much important here? Why? Because अगर ये enzyme deficient होगा, if this homogentisate enzyme is deficient, then of course this reaction will not occur. And if that reaction does not occur, homogentisate levels will go up in the blood. And not only in the blood, homogentisate level is not only increasing in the blood, what is going to happen? That will start excreting in the urine also. Now collect that urine sample and keep that urine sample without disturbing for some time. Then you will observe that, yes, my dear friends, you'll observe that the color of the urine, which is slightly yellowish in color, will change to black color. And that disease is known as black urine disease or alkaptonuria. And this has been already asked in your FMG exam last year, sir, 2023. So please write down there as clinical correlation, and the disease name will be alkaptonuria. Alkaptonuria, it is also known as black urine disease. Now, black urine disease, what is the enzyme deficient in this one? The enzyme deficient will be homogentisate oxidase. Alkaptonuria or black urine disease, the enzyme deficient will be homogentisate oxidase. Homogentisate oxidase. And what is the clinical feature of this one? Yes, please write down there. The clinical feature I'm writing here, sir. Urine on long standing, comma. This word was used in your exam, sir. Long standing. Long standing मतलब हम क्या करेंगे? We are going to just collect the urine sample and keep it without disturbing for some time, sir. I mean to say we are not going to add anything, no reaction there, nothing to add, nothing to remove, कुछ नहीं करेंगे. We are just going to take the urine and keep it for some time. And then you'll observe that urine on long standing, you'll observe that the color of urine changes to black color or coke colors. The urine on long standing, the color of the urine will change to what, sir? Changes to black color or coke color. And that is why this disease is known as alkaptonuria. Alkaptonuria or black urine disease. And one more clinical feature that you have to remember here in this is what is going to happen, sir. Ochronosis bhi hota hai. Now, what is the meaning of ochronosis here? What is going to happen is this substance homogentisate will not only just get excreted from our body in urine. Remember, that is also getting deposited in the connective tissues in our body. And even those connective tissues also, it is converting to black color. So it is converting the urine to black color as well as the connective tissues to black color. So therefore, that condition is known as what, sir? Ochronosis. Acha, how you will be able to appreciate that one? आप कैसे देखोगे clinical condition में? In a clinical condition in the patient, how will you observe that one? So please remember that you will be able to see in the pinna. In the pinna, this is actually the elastic cartilage, or ये cartilage में deposit होगा, and this is going to become like blackish in color. ठीक है ना? तो एक कार्टिलेज में डिपॉजिट होकर ना उसको ब्लैक कर देगा, इट इज गोइंग टू कन्वर्ट इनटू ब्लैक कलर, और दैट कंडीशन इज नोन एज व्हाट, ओक्रोनोसिस। ठीक है, तो व्हाट इज ओक्रोनोसिस सर? डिपोजिशन ऑफ homogentisate in connective tissues like cartilage or sclera, etc. In the cartilage or in the sclera, the sclera of the eyeball here, वहां पर भी वो डिपॉजिट होता है, एंड दैट कंडीशन इज नोन एज व्हाट सर? ओक्रोनोसिस कहते हैं, दैट इज नोन एज ओक्रोनोसिस। क्लियर? एंड बाय दिस वी आर डन विद वन मोर डिसीज कंडीशन हियर, and by this, my dear friends, we are actually done with one of the very, very big topic here, that is metabolism of aromatic amino acid completed here, sir. Oh my God. So tryptophan, phenylalanine, and tyrosine, these three amino acid metabolism is a big, big one. If this is done, actually majority of the protein metabolism is completed here, sir. So we are done with a big, big metabolism here, sir. So on the screen, you are able to see the pictures of ochronosis. Ochronosis there, sir. Accumulation of homogentisate in the cartilage as well as the sclera and in the palm, everywhere you can see that one. That is known as ochronosis, sir. We are actually done with the metabolism of aromatic amino acids, sir. Let us get started with the metabolism of aliphatic amino acids, sir. Now, please recall in your mind, first of all, sir, yesterday we have seen the classification. Aliphatic amino acid. Yes, my dear friends, aliphatic amino acids, please recall what are the names of the aliphatic amino acid. Glycine, alanine, valine, leucine, isoleucine. These are the aliphatic amino acids. We are going to learn about them one by one. So first, let's get started with the glycine here, sir. Now, glycine is very much important, sir. Many questions are asked from here in your FMG exam previously. Question number one, we all know that it is the smallest amino acid. And not only smallest, remember it is the simplest amino acid, sir. It is the smallest and simplest. And one more thing, remember, glycine is the most abundant amino acid in collagen protein, sir. Even this question is also asked in your exam, sir. तो अगर आप collagen protein देखोगे ना, if you look at the structure of collagen is obviously a protein. It is made up of amino acid, many amino acid, and it is given very clearly that every third amino acid will be glycine. It is most abundant amino acid in collagen, and glycine is the one which is responsible for the bends and flexibility in protein chain. What is the meaning of this one? Agar मान लो कि you are looking at the long chain of proteins, so there's a long chain of amino acid, and suddenly there is a bend. So wherever there will be a bend, at that bend, you'll be able to find what's a glycine. So because it is the smallest one, it will be able to easily accommodate in the bend of the protein chain. So protein chain bends or flexibility is due to which amino acid? Glycine. So first you remember there's three important MCQs here, sir. Now, after learning these MCQs, now write down there as the functions of glycine, sir. What are the functions of glycine? Number one, glycine will be actually helping in heme synthesis. Glycine is required for heme synthesis, sir. Kaise? How it is required? Remember, in heme synthesis, the very first reaction will be succinyl-CoA will combine along with glycine to form delta amino levulinic acid, delta ALA, and that will be finally undergoing many reactions to form what's a heme. So, my dear friends, remember, my dear friends, hello. Okay, heme synthesis we are going to learn separately in the hemoglobin topic, sir. Heme synthesis we are actually going to learn in the hemoglobin topic separately. Okay. There is a separate topic. I'm going to teach you in detail about the heme synthesis pathway. But for now, my dear doctors, for now, remember, okay, Look at the very first reaction there. In the very first reaction, you are able to very, very clearly see here, yes, sir, glycine is required for heme synthesis. Now, please focus only on glycine. So I'll teach you that separately. Don't worry about that one. We are going to learn the pathway. Glycine is required for heme synthesis, number one. Number two, the second thing is glycine is also required for purine ring formation. Glycine is also required for purine ring formation, sir. I hope you all know the purines, sir. Purines will be adenine and guanine. Adenine and guanine. And remember, purine ring will be actually, it will be a double ring structure. And this double ring will be actually heterocyclic, sir. I'll tell you what is the meaning of this one. Now, in the purine ring, if you look at the structure, one of the ring will be hexagonal ring, like six-sided. Another ring will be the pentagonal, sir. Please draw it slowly, aram se. Don't draw both of them hexagonal. One is hexagonal, six-sided. Another one is pentagonal, sir. Okay, hexagonal, and then the another one will be the pentagonal, sir. There'll be like double ring structure. And number two, second thing that you have to remember is that it is heterocyclic. Now, what is the meaning of homo? Homo means it will be the same, whereas hetero means it will be made up of different atoms, sir. That is not made up of all the similar atoms. For example, I think you people remember in the lower classes you studied about benzene ring. And in benzene ring, the entire ring is made up of only the carbons only. So that is homocyclic. But here it is heterocyclic. It is made up of both carbon as well as my dear friends, remember, it is made up of nitrogen, sir. Carbon and nitrogen. Now, in this in this purine ring, there will be carbon number 4 here, and then there will be carbon number 5 here, and then there will be nitrogen number 7 here. And this carbon number 4, carbon number 5, and nitrogen number 7, this will be contributed by glycine. This is actually given by glycine, sir. So glycine is required for purine ring formation, sir. Not pyrimidine, only the purine ring formation. Acha. Don't think that Azam sir is not teaching the entire ring here right now. Yes, I'm going to teach the entire ring also, but not now. Yes, we'll be studying in the DNA and RNA topic, sir. DNA and RNA topic, okay? Next one. Function number 3, glycine is also required for glutathione formation. Glycine is also required for what, sir? Glutathione formation. Okay, now remember, glutathione is actually a tripeptide, sir. Now, what is the meaning of tripeptide here? Tripeptide means that it will be actually made up of 3 amino acids. And your examiner wants you to remember all 3 amino acids, sir. Your examiner wants you to remember all the 3 amino acids. So, how to remember all the 3 amino acids, sir? How we are going to remember all the 3 amino acids? It's very, very simple. It's there in the name itself. So, remember glutamate. And I hope you know a little bit of chemistry. Whenever you're going to come across the word of thio, the word as thio, okay, now, thio means what's a sulfur. And here there will be sulfur containing amino acid, that is cysteine. And then after that, the last one will be ONE1. The first one amino acid will be glycine. Oh, yes, sir. Yes, my dear friends, all the 3 amino acid name is written in the name itself, sir. Okay. So glutathione formation के लिए भी glycine चाहिए. Then apart from that, function number 4, glycine is also required for the formation of creatine. So creatine formation. And again, creatine is also made up of 3 amino acids. So 3 amino acids you have to remember. Remember with the mnemonic as GAM. G stands for glycine. A stands for RG9. And M stands for methionine, sir. Glycine and RG9 and methionine. These 3 are the one which are going to contribute for the formation of creatine, sir. Okay, so glycine is required for the formation of 4 important substances. Number 1, it is required for the formation of heme. Number 2, glycine is required for the formation of purine ring. And number 3, glutathione, as well as, my dear friends, creatine. So these are the four important substances glycine is forming. So please remember that very carefully. Then number 5, glycine is also acting like a neurotransmitter. Glycine is also acting like a neurotransmitter. I think आप लोग ने ना physiology में पढ़ा होगा, ठीक है ना? In the physiology, I hope people have learned about the neurotransmitter that is like, excitatory and inhibitory neurotransmitters. Sir. I think you have learned about the inhibitory neurotransmitters. So one of the inhibitory neurotransmitter will be GABA, और आपने सीखा होगा GABA के अलावा, like apart from GABA, there will be even glycine. So GABA, glycine, all this will be actually your inhibitory neurotransmitter. That is also acting like a neurotransmitter, sir. Then number 6, and one more important function of glycine. Glycine is also required for conjugation of bile acids. Conjugation of bile acids. That's all. These are all the functions of glycine that you have to remember. Yes, of course. Is it okay? Conjugation of the bile acids of glycine. Okay? ये हो गया glycine completed. Done. Now we are learning the metabolism of which amino acids, sir? Aliphatic amino acid. And in aliphatic amino acids, remember, glycine done. After glycine, the next one that we are going to learn is alanine. And the good news here is that in alanine, neither I have to teach you anything, nor you have to learn anything, sir. Why? Because alanine will be involved in Cahill cycle. In fact, the another name of Cahill cycle is glucose alanine cycle. So now you only tell me, did we study about this cycle in carbohydrate metabolism? हमने पढ़ा है कि नहीं पढ़ाया सर? Yes, we do have learned about this one. है ना? है ना? So glycine, alanine is required in the Cahill cycle, and Cahill cycle already we have learned in the along with the Cori cycle. Very good, Dr. Vishal. So we don't have to learn it like again and again. Already we have completed that conceptually before itself, sir. Okay, na? Then after that, the next one which are remaining will be your branched chain amino acids, BCAs. And what are the branched chain amino acids, sir? Number 1, they will be valine, and then you'll be having leucine, and then there will be isoleucine. Valine, leucine, and isoleucine, sir. Is it okay? These 3 are the branched chain amino acid, and this topic is very much important. You can expect a question from here in your next exam, sir. Now, what you have to remember here is valine, leucine, isoleucine, these 3 amino acids will undergo metabolism. No need to learn the pathway. And it is going to finally form some end products, and no need to even learn the end products also, sir. Valine, leucine, isoleucine, they will undergo certain reactions. So वो pathway भी पढ़ने का आपको जरूरत नहीं है, ठीक है ना? And then finally products are formed. No need to learn that one, sir. Now during that pathway, my dear friends, there is one enzyme. Only that one enzyme you have to remember. Okay, what is that one enzyme? Let us make the name slowly. These are all like branched chain amino acid. Na? So इसीलिए the name will be branched chain. And then I told you yesterday, after removing the ammonia, what is left out? Keto acid. So already from this amino acid, already ammonia is removed. Do you remember our overview, sir? है ना? तो already वहां से ना ammonia is removed here, and then what is left out here, sir? Keto acid. And then, my dear friend, यहाँ पर oxidation हो रहा है, and you know what is the meaning of oxidation here, sir? Removal of hydrogen. So dehydrogenase. So you have to remember the name of the enzyme here in this manner, sir. Okay? Is it okay? So branched chain keto acid dehydrogenase. Branched chain keto acid dehydrogenase, sir. Repeat again. So what is the name of the enzyme here, sir? Branched chain keto acid dehydrogenase. And this enzyme name, I don't know how you remember. I remember this one as BCKD. So please remember this enzyme as BCKD. Branched chain keto acid dehydrogenase. Very, very, very important one, sir. Why? Now, why it is important is why? Because first of all, this enzyme also requires what, sir? Coenzymes for functioning. And not even like one or two, totally 5 coenzymes are required. Number one, there will be lipoic acid. Number two, there will be thiamine pyrophosphate. Number three, there will be coenzyme A. Number four, there will be NAD plus. And number five, there will be FAD, sir. So these 5 coenzymes are required. I hope you are able to recall. Somewhere else also we have learned about these 5 coenzymes, sir. कहीं और हमने पढ़ रखा है about these 5 coenzymes, sir. So we learned about these 5 coenzymes. Yes, link reaction. Very good, Dr. Poonam. Very nice. Yes, pyruvate to acetyl. Very good, Dr. Najib. Very nice. So 5 coenzymes required. Now the most important thing, yes, in some people there will be deficiency of this enzyme. So therefore, when this enzyme is deficient, it will lead to a disease known as maple syrup urine disease. MSUD, maple syrup urine disease. Now you all know very well in biochemistry, whenever you're learning about any disease, first of all, make a habit of writing enzyme deficient. Now what is the enzyme deficient here, sir? BCKD, branch chain keto acid dehydrogenase. Branch chain keto acid dehydrogenase. Okay, maple syrup urine disease, this is the enzyme deficient. Now, what is the clinical feature of this one here? अरे यार ये बताओ, like why did they give the name as maple syrup? Maple syrup यहाँ पर इसका नाम क्यों रखा गया है? The disease name is maple syrup. तो इसका नाम ऐसे क्यों रखा गया है? Why? Why? Because remember, in the urine, the smell will be like the smell of the maple syrup, and that is like burnt sugar-like smell. The smell will be like burnt sugar. So you have to remember even this one also. Burnt sugar-like odor in urine. So this is known as what, sir? Maple syrup urine disease. But yes, okay now. So each and every point in this one is very much important here. Now what you do is please add a note to this. A note करके लिखिए. And in the note, please be very, very careful here, sir. यहाँ देखिए. Maple syrup urine disease. Suppose in the exam, I'm asking you the question, what is the enzyme deficient? What will be your answer, sir? The enzyme deficient will be BCKD. Branch chain keto acid dehydrogenase. Okay. But if I ask you what is the defective mechanism here, which mechanism will be defective, then your answer should be what, sir? Decarboxylase. Actually, it is a very big enzyme having like 5 complexes, 5 coenzymes are required in that one. And that is the reason why you remember, like there are so many different reactions are taking place. So main defect here, the defect here will be what here, sir? Yes, decarboxylase. So if in the option, like, it's very simple here. In the option, for example, agar wahan par na dehydrogenase nahi hai, if in the option you don't find dehydrogenase, then your answer should be what, sir? Decarboxylase. If there is no dehydrogenase in the option, then go for decarboxylase. Done. And by this, we are done with all the aliphatic amino acids. Glycine, alanine, valine, leucine, and isoleucine. That's it. Sure. Aromatic हो गया और aliphatic भी हो गया, sir. Now, please write on the heading there as metabolism of basic amino acids, sir. Now, please recall what are the basic amino acids, sir? Basic amino acids are lysine, arginine, and histidine. So first of all, let us learn about the histidine. And remember, histidine is the one which is going to form histamine. I think you have come across this histamine a lot of time in your pathology. Histamine, serotonin, that vasodilators and all those things. So histidine is the one which is going to form histamine. But in your exam, they have asked how it is going to form that, sir. So remember, it is formed by decarboxylation. Next one. The next one that we have to learn is arginine. Or arginine to aaj hi humne padha tha. Today only we have learned in the very beginning, first topic, arginine will be involved in the urea cycle. The first thing is it is involved in urea cycle. And in fact, arginine was the one which will break down to form urea and ornithine. Urea cycle में आएगा. And the most important one, the very, very important one, जो आपके MCA एग्जाम में repeatedly पूछा गया है कि arginine is the one which is required for the formation of nitric oxide. The formula will be NO, nitric oxide formation. Very good. And for this, you also have to remember the reaction. In our body, we'll be having L-arginine, and L-arginine is the one which is going to form nitric oxide, that is NO, in the presence of which enzyme? Remember NOS, nitric oxide synthase, NOS. So arginine is the one which is going to form the nitric oxide. Is it okay? हो गया जी, done. So by this we are done with what here, sir? We are also done with what? The basic amino acids are also completed here, sir. Basic amino acids are also done, sir. So yeah, actually, tell me one thing here. Are you all able to understand something here in the proteins topic, what we are learning? Like where we have to focus actually in the proteins topic. कुछ समझ में आ रहा है आपको? So your focus as a student should be like, which amino acid is actually going to form which important products. That is something important. Nitric oxide will be derived from where? Arginine. Histamine is formed from where? Histidine. Melanin is formed from where? Tyrosine. So in this manner, you actually have to remember, which important substance is actually formed from which amino acid. So amino acids are going to form certain important products. So you have to remember those important products themselves. And yes, of course, the clinical correlations also very much important, sir. Alkaptonuria, phenylketonuria. These are like commonly asked questions in the exam, sir. Now welcome to the last and final concept in the proteins topic, sir. We'll do this concept and the proteins topic will be completed here. So protein metabolism will be done. And it is very much important concept. And I'm now going to teach you in detail and very, very clearly about the megaloblastic anemia, sir. And once you learn this megaloblastic anemia, sir, you are done with the proteins topic. Then we'll start with the lipids topic, sir. और ये बहुत इंपॉर्टेंट है इन योर एमसीए एग्जाम दे आर गिविंग दिस वन एज अ क्लिनिकल क्वेश्चन सर मेगलोब्लास्टिक एनीमिया so you have to know in depth about that one. Okay, now I hope you all know very well megaloblastic anemia will be either due to deficiency of vitamin B12 or it is due to deficiency of folic acid, sir. That is vitamin B9. And please observe my English very carefully. I'm writing there as or. Now, what is the meaning of this one? The meaning here is that megaloblastic anemia will either occur due to deficiency of vitamin B12 or the folic acid. It's not necessary that both of them has to be decreased there, sir. Okay, either of the one will be actually deficient. Either of the one will be decreased, sir. Done. Are you understanding here, sir? So therefore, what is going to happen now? Now our headache will increase more now. We'll be having like more challenge right now, sir. I mean to say, what is the more challenge right now? What is more challenging right now? Suppose from now onwards, a patient comes to you with megaloblastic anemia, sir. The moment any patient comes to you with megaloblastic anemia, now what you have to do? अब आप क्या करना पड़ेगा आपको? First of all, you have to do the differential diagnosis, sir. कि vitamin B12 deficiency है या folic acid deficiency है. We have to do the differential diagnosis that whatever patient is sitting in front of you, that patient is having the deficiency of vitamin B12 or folic acid. Now, how will you do the differential diagnosis, sir? अब इसको हम कैसे हम differentiate करेंगे? And for that, a little bit of biochemistry is required, sir. So I'll teach you like two diagrams. In two diagrams, everything will become like crystal clear. Okay. So what you do is leave like two lines here. एक दो line यहाँ पर ना छोड़ दो. We'll come back to this later on. First, let us learn the two diagrams here, sir, the two concepts. Now you write down the heading there as metabolism of sulfur-containing amino acid. Do you all remember sulfur-containing amino acid? The sulfur-containing amino acid, sir. Cysteine and methionine, of course. And now, just before the break, I taught you one reaction where norepinephrine is getting converted to epinephrine, sir. Did I teach you this reaction before the break? हाँ, very good, Dr. Rekha. Very good, Dr. Vandana. Very nice. Norepinephrine to epinephrine is a methylation reaction. And who is the methyl group donor? SAM. So SAM will give the methyl group. And after giving the methyl group, it is going to become SAH, S-adenosyl homocysteine. Now, my dear doctors, remember that. Don't think that in our body, this is the only one methylation reaction. No, 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 no. In our body, we have like many methylation reactions. Guys, are you listening to me? 5 minutes, this concept will be like crystal clear. Everyone, please be with me. So, who is the methyl group donor here, sir? SAM. SAM will give the methyl group. But now the problem with SAM is that in our body, there are many methylation reactions, sir. बहुत सारे हैं. There are so many methylation reactions. And wherever methyl group is required, वहां पर ना SAM पहुंच जाता है. SAM will always be available to give the methyl group. Now it makes sense, common sense, के wherever you require the methyl group, SAM is giving that, but it is not like just only utilizing SAM. है ना? We have to even regenerate the SAM also. ऐसा नहीं कि बस उसको हम यूटिलाइज करते जा रहे हैं. We have to even regenerate the SAM also, right? So how to regenerate? Simple. SAM has given the methyl group. अब क्या करना पड़ेगा? If you want to regenerate this back, you need to add the methyl group, sir. Simple. You have to add the methyl group. ठीक है ना? Just keep that logic in your mind. So now what is going to happen? S-adenosyl homocysteine will further break down to form homocysteine. S-adenosyl homocysteine will break down to form homocysteine. Okay. And now this homocysteine is going to react with methyl tetrahydrofolate. This homocysteine is going to react with what, sir? Methyl tetrahydrofolate. नाम में लिखा है methyl group है sir. It is having the methyl group. So our condition is satisfied here. We have to add the methyl group. Now after adding the methyl group, sir, मान लो कि ये methyl group यहाँ पर add हो गया है. What is left out? Only tetrahydrofolate. And after receiving the methyl group, homocysteine is going to become what, sir? Methionine. ये क्या बन जाएगा? Methionine. So methyl tetrahydrofolate, after adding the methyl group there, homocysteine is going to become what, sir? Methionine. And then after that, what is the name of the enzyme here? Of course, the name of the enzyme will be methyl transferase. And what is the complete name of the enzyme? Homocysteine methyl transferase. And I call this one as HMT. Homocysteine methyl transferase. And this is the enzyme which will require vitamin B12 as a coenzyme. तभी तो मैं आपको पढ़ा रहा हूँ. That is the reason why I'm teaching you here, because we have to learn about what, sir? Megaloblastic anemia, vitamin B12 deficiency. तो vitamin B12 यहाँ आता है, ठीक है ना? And after that, this methionine is actually getting converted to what, sir? S-adenosyl methionine. SAM. Methionine is going to become what, sir? S-adenosyl methionine, sir. Clear with this one? So first topic completed here, sir. One diagram is done. Are you all clear with this one here? So I promised you like 2 diagrams here, so 1 diagram is done. And my dear friends, remember, don't think like it is such a vague reaction or something like that. Nothing. अगर आप ध्यान से देखोगे, I taught you only and only one reaction here, sir. HMT, homocysteine methyl transferase. That's all. That's all. Okay. बाकी का सारा, all these things is actually your background story. It's all background story. Just focus that I have taught you only one reaction here, sir. मैंने बस आपको एक reaction पढ़ाया, sir. Only and only one. Sure. Now, once you are done with this one here, now the next one, propionic acid pathway. Please write on the heading as propionic acid pathway. Now, this is also very simple. Only 2 reactions we have to learn. Now, in biochemistry, there is something very famous known as vomit, sir. Vomit करके होता है. Now, what is this vomit here, sir? Vomit में ना V stands for valine, O stands for odd chain fatty acid. M stands for methionine. I stands for isoleucine. And T stands for threonine, sir. Okay. So in biochemistry, there is something very important known as vomit, sir. Okay, now. Now, what is the meaning of vomit here? Vomit में क्या होता है? Sir, V for valine, O for odd chain fatty acid, M for methionine, and then I for isoleucine, and T for threonine. Now, what is the importance of this one, sir? The importance of this one is that all these 5 amino acids, when they undergo metabolism, the end product will be the same, sir. All that 5 amino acids, when they undergo metabolism, their end product will be the same. And their end product will be the propionyl CoA. That is why it is important to learn them together. So all the 5 amino acids, like, valine, methionine, isoleucine, and all of them, when they undergo metabolism, the end product will be what, sir? Yes, propionyl CoA. Now, this propionyl CoA, first of all, it is getting converted to methyl malonyl CoA. I want your full attention here, sir. Last 2 reactions, but extremely important. Extremely important, sir. Methyl malonyl CoA. I remember that as MMC. Methyl malonyl CoA, MMC. Now, how is this propionyl CoA getting converted to methyl malonyl CoA? By addition of carbon in the form of carbon dioxide. So if you're adding carbon, what is the name of the enzyme? The name of the enzyme will be carboxylase. And what is the full name? The full name will be propionyl CoA carboxylase. Add the name of the substrate along with that. That is propionyl CoA carboxylase. Propionyl CoA carboxylase. और अब क्या होगा? Now what is going to happen? Remember, propionyl CoA carboxylase is the one which will be requiring a coenzyme, and that coenzyme will be biotin. Oh my God. कुछ याद आ रहा है? Are you able to recall something? Biotin, I told you very clearly that on day 1, yes, very good, Dr. Alisha, very good. Biotin is required for how many enzymes in the entire biochemistry? Remember, biotin is required for 3 reactions in the entire biochemistry. And this is the second one that we are learning today, sir. I hope you are making the list of that somewhere. Please make a list of that somewhere, sir. So propionyl CoA carboxylase requires what? Biotin. And now what is going to happen? Methyl malonyl CoA will get converted to succinyl CoA. How it is getting converted to succinyl CoA? Yes, with the help of an enzyme known as mutase. And what is the full name? साथ में substrate का नाम लगा दो. तो methyl malonyl CoA mutase. MMC mutase. Methyl malonyl CoA mutase, MMC mutase. And this enzyme requires vitamin B12 as the coenzyme. Oh, vitamin B12, megaloblastic anemia में आएगा. Keep an eye on that. Keep your focus on that one. Understood? That's all. So as I promised you, I'll give you just 2 reactions here, sir. So the 2 reactions are done. And this topic is also completed here, sir. ठीक है ना? बस 2 reaction आपको यहाँ सीखने हैं, and both the reactions are very much important. Why? Because one of the reaction is requiring biotin as a coenzyme. Another one will be requiring vitamin B12 as a coenzyme here, sir. So both of them are very much important here. Done? So this diagram is also done. This diagram is also done. And now welcome to our condition right now, sir. जो हमने जहां से कहानी शुरू की थी, sir. We started the story from megaloblastic anemia. Are you ready? Are you ready? Now, welcome here right now, sir. Megaloblastic anemia. See, megaloblastic anemia will occur either due to deficiency of vitamin B12 or the folic acid. Let us take case number 1. For example, there is a patient suffering with vitamin B12 deficiency. Now, remember, my dear doctors, from today onwards, The moment you listen vitamin B12 deficiency, two enzymes should come in your mind, sir. One enzyme will be HMT, homocysteine methyltransferase, and another enzyme will be the MMC mutase. From now onwards, from today onwards, remember that the moment you listen vitamin B12 deficiency, these two enzymes should come in your mind, sir. HMT and this one here. अब आप मुझे बताओ, हेलो जी, अगर मान लो कि विटामिन B12 डेफिशिएंट है, अच्छा, तो will this reaction take place? This enzyme will work? No. Will this reaction will occur? No. Therefore, if this reaction is not taking place, then what is going to happen? Homocysteine levels will increase because it is not utilized. So in vitamin B12 deficiency, number one, what is happening here, sir? Homocysteine levels will increase, number one. Number two, vitamin B12 is also required for this reaction also. If vitamin B12 is deficient here, this reaction is not taking place. So therefore, even methyl malonyl CoA levels will also increase. अच्छा, तो homocysteine भी बढ़ेगा and even methyl malonyl CoA will also increase. Both of them will be increased in vitamin B12 deficiency. Now let us cross-check that with the folic acid deficiency. Now, man lo you're having another patient suffering with megaloblastic anemia, but in another patient, patient number 2, it is due to the deficiency of which one? Folic acid. Vitamin B9 deficiency. So just now I said you here, my dear friends, methyl tetrahydrofolate. And right from day one, I'm telling you the same basics. Ic acid will form 8 salt. Ic acid will form what, sir? 8 salts, sir. ठीक है ना? So nothing but folic acid is going to form what? Folate. So this is coming from where only, sir? Methyl tetrahydrofolate is coming from where? Folic acid only. Vitamin B9. Now what is the problem that we are having? The problem is folic acid is deficient. If this is deficient, then there'll be no methyl tetrahydrofolate. If this is not there, again, this reaction will not occur. If this reaction doesn't occur, again, homocysteine levels will go up, sir. So therefore, my dear friends, remember, even in folic acid deficiency, also what is going to happen? Homocysteine levels will increase. Even in folic acid deficiency, also homocysteine level will increase. Now, my dear friends, oh my God, confusion increased more now. Confusion increased more now. Now in both of them, homocysteine is increased. How to do the differential diagnosis? Please look at this reaction number 2 here, sir. Yeah, well, this one reaction. Now tell me, in this reaction, do you have any role of folic acid? No. If there is no role of folic acid, will methyl malonyl CoA levels increase, decrease, or change, something like that? No. उस पे फर्क ही नहीं पड़ेगा. The levels are not going to either increase, decrease, something like that. No change in that one. So therefore, there'll be no change in methyl malonyl CoA here, sir. That means now from today onwards, in the hospital, when you start practicing, a patient comes to you with megaloblastic anemia. What is the final conclusion? You are going to do a blood test. And when the report comes of the investigation, blood test, in the report, if only homocysteine elevated, but clearly likha hoga, so it will be written very clearly. If only homocysteine elevated, then yes, sir, you're suffering with folic acid deficiency. If both homocysteine as well as methyl malonyl CoA is elevated, yes, sir, you're suffering with vitamin B12 deficiency. That is how you are going to treat the patients. You are going to diagnose that person. Understanding here? So this is how you're able to identify megaloblastic anemia due to vitamin B12 deficiency or folic acid deficiency. Is the concept very clear now? Is the concept very clear right now, sir? Yes, Dr. Rahul Singh, even that will be going there also. Yes, in the urine also. So in this manner, what happened, we are done with megaloblastic anemia also. We are done with sulfur-containing amino acid also. We are also done with vitamin B12 requiring enzymes. 2 enzymes require vitamin B12. And we are also done with the biotin requiring enzyme. Like so many things are completed at one place. So many things are completed here, sir. And in one of the exams, they have also asked in depth here, my dear friends, okay, this one, odd chain fatty acids, sir. What is the end product of odd chain fatty acid? The end product is propionyl chloride. That should be your answer. Even that question was also asked. And nowadays, people are not at all learning in a conceptual manner. Everyone wants that last 2 hours of rapid revision session on the YouTube. Sir, and on to that, अच्छा वो 2 घंटे पढ़ेंगे और उसके बाद में ना मुझे मैसेज भी करते हैं, सर ये मतलब रैपिड रिवीजन ओके ना सर, मतलब बायोकेमिस्ट्री मैंने कभी नहीं पढ़ा है सर, ये 2 घंटे पढ़ लेंगे तो हो जाएगा सर। अच्छा भाई, दे वांट अ कंफर्मेशन ऑल्सो फ्रॉम मी। अरे भाई साहब, दैट रैपिड रिवीजन इज एक्चुअली फॉर हायर टॉपिक्स, मतलब वो तो पढ़ के ही जाना है, यू हैव टू स्टडी एंड गो टू द एग्जाम। इट मीन्स दैट वन, like quick revision of the subject तो पढ़ना पड़ेगा ना सर, at least once you have to learn the subject, right? That is why they have given the word as revision. For revision, once at least you have to vision, understanding। बिना एक बार भी विजुअलाइज किए, बिना एक बार ही सब्जेक्ट पढ़े, डायरेक्टली बस रिवीजन चाहिए, वो भी बस 2 घंटे, मतलब 2 घंटे बायोकेमिस्ट्री हो गया। लोग पागल हैं जो एक एक साल के लिए पढ़ते हैं, और हम यहाँ पागल है जो बैठ के 3-4 दिन बायोकेमिस्ट्री पढ़ रहे हैं और 2 घंटे में हो जाएगा भाई, पढ़ लो भाई मेरा। तो दे विल बी आस्किंग द क्वेश्चंस फ्रॉम द, मतलब इन द मिडिल एंड देन, और अच्छा ये लोग वही होते हैं फिर जो 2 घंटे पढ़ाई करते हैं और उसके बाद में ना यस आफ्टर दैट दे विल बी राइटिंग आफ्टर द एग्जाम, अरे भाई सर के नोट से नहीं आया, कौन से नोट से, रैपिड रिवीजन वाले। सही है भाई, अक्ल ही नहीं है। एंड द सेम थिंग विल बी कैरिड ऑन टू द नेक्स्ट बैचेस आल्सो, दे विल नॉट बी फॉलोइंग प्रॉपर्ली, पता नहीं, वो लोग कंफ्यूज रहते हैं और दूसरों को ज्ञान भी देते रहते हैं, भाई यहाँ से आता है वहां से नहीं आता है, यार सारी बातें छोड़ो, सिट डाउन रिलैक्स, आराम से बैठो और आराम से अपनी तैयारी करो। डोंट गेट इन्फ्लुएंस विद एनी अदर आंसर, उनका ओपिनियन उनके साथ में, बोलो थैंक यू, आपका ओपिनियन आपकी जिंदगी, आप जो करना चाहो करो, मेरी जिंदगी मेरी है, मेरे हिसाब से जीने दो, ठीक है ना। तो डोंट ट्राई टू जस्ट गेट इनटू दोस काइंड ऑफ ट्रैप्स एंड कंफ्यूजन्स, आराम से लर्न द सब्जेक्ट क्लियरली एंड दैट विल बी हेल्पिंग यू लॉट सर, ठीक है ना। और इतना कुछ डिफिकल्ट है भी नहीं यार, पता नहीं। खैर खैर जो भी हो, वट एवर इट इज। चलिए जी आगे बढ़ेंगे, शैल वी गो अहेड नाउ। एक काम करता हूँ सर मैं, बाय दिस व्हाट हैपेंड इज द प्रोटीन्स टॉपिक इज डन हियर। तो बिग टॉपिक कार्बोहाइड्रेट डन विद ऑल द अपडेट्स, गैलेक्टोसीमिया, classical galactosemia, all the clinical conditions and all glucose transporters. So we are done with the carbohydrate metabolism, then we are done with the protein metabolism right now. These two big topics are done. Now next what we'll do is we'll do one small topic here, sir. That is hemoglobin topic, sir. Hemoglobin topic यहीं पर पढ़ेंगे तो थोड़ा सा ना easy हो जाएगा. So please write on the heading there as hemoglobin, sir. Now we all know that in hemoglobin there'll be like two parts. One will be the heme part, another one will be the globin part, sir. और आपको इतना तो पता होगा कि out of these two, globin will be the protein part, whereas heme will be the non-protein part, sir. Hemoglobin will actually have like two parts, the heme part and the globin part. Globin will be actually the protein part and heme will be the non-protein part, sir. ठीक है ना? Now in this, I don't have to learn about the structure of globin. Why? Because globin is protein and we have just now learned proteins are made up of what, sir? Amino acids. Proteins are made up of amino acids only. We know that very well. So we don't have to learn the structure of globin, but yes, we have to learn the structure of heme right now. It is a non-protein part. So let us see the structure of heme first of all, sir. Structure of heme, sir. Now in heme molecule, first of all, there will be a pyrrole ring. अब ये pyrrole ring कैसे नजर आता है सर? It will be pentagonal, five-sided. At one of the end, you will be having nitrogen, and remaining all the four ends will be having carbon, 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 and the carbon here. That's a pyrrole ring, sir. And don't think there is only one pyrrole ring here. Totally, there will be like 4 pyrrole rings, sir. And they are so beautifully arranged that all their nitrogens will be directed towards the center. In the 4 pyrrole rings, the nitrogens will be directed towards the center. Okay. Now, all the 4 pyrrole rings are joined together with the help of these methylene bridges. All the 4 pyrrole rings will join together with the help of these methylene bridges. So therefore, what is going to happen? Now it is looking like a big ring-like structure. And this big ring-like structure will be known as what, sir? Protoporphyrin. So all the 4 pyrrole rings will join together And all of them will join together to form a big ring-like structure, and that is known as what's a protoporphyrin. Protoporphyrin. Now, in this protoporphyrin, in the center, you're going to add the iron in the form of ferrous, Fe+2, sir. And once you add the iron in the center, that's all. It is known as what, sir? Heme. इसी को हम क्या कहते हैं? Heme कहते हैं, sir. So please add iron in the center, Fe+2, and that is going to become what, sir? Heme, sir. इतना आसान. It is so simple. So first you'll be having like 4 pyrrole rings. Join them together. It will be protoporphyrin. And then add iron in the center. It is going to become the heme, sir. हो गया. Heme का structure done. Heme completed here, sir. Okay. Now, यार ये बताओ, how many globin chains do you have in the hemoglobin? There will be like 4 globin chains. So imagine like you're having like 4 globin chains, the protein chain, and there will be 2 alpha and then 2 beta chains. इसीलिए when you learn in pathology, it will be writing as alpha to beta 2. So alpha to beta 2. Now, one of the problem with many of the students is that okay, they will be thinking in hemoglobin there is only one heme. No. Each and every globin chain will be attached with one heme molecule. 4 pyrrole ring. And then join them together, protoporphyrin. और center में लगा दो iron यहाँ पर sir, Fe+2. So this is like a heme molecule here, है ना? But students are thinking there is only one heme molecule. No. Each and every globin chain will be attached with one heme molecule. So कुल मिलाकर how many heme molecule do you have, sir? 4. So even you'll be having like 4 heme molecules. And this together is known as what, sir? Hemoglobin. So from today onwards, at least remember this schematic diagram, sir. Remember, it is not a like actual diagram of the hemoglobin. Hemoglobin is actually having a very complicated structure. It's not like the actual hemoglobin. ये हमारे पास याद रखने के लिए है, sir. ठीक है ना? So this one is like your HB, hemoglobin. So there'll be how many globin chains? 4. And all the 4 globin chains will be attached with one heme molecule, sir. Hemoglobin. Sure. Structure is like crystal clear now, so very clear now. Now, once you are done with the structure, let us again learn the synthesis of heme right now, sir. Arey, be very careful here. I'm not telling synthesis of hemoglobin, sir. See, don't write this once again. Please don't write. Hemoglobin होता है ना? In hemoglobin, globin will be the protein part, but heme will be the non-protein part. Now, we don't have to learn the formation of globin. No. Globin का formation हमको पढ़ना ही नहीं है. Why? Because globin is a protein part. And proteins are made up of what, sir? Amino acid only. So we don't have to learn the formation of globin. We just have to learn the formation of the heme, sir. Heme का formation हमको पढ़ना है. So please write down there the heading as synthesis of heme. Heme synthesis. और मैंने आपको अभी बताया था, just after the break I taught you, succinyl CoA will combine along with glycine to form delta amino levulinic acid, which is known as delta ALA. Succinyl CoA combines with the glycine to form what's a delta ALA. And this delta ALA will first get converted to porphobilinogen. Porphobilinogen, sir. In short, we'll write it as PBG. And first, I would request you people to learn this reaction. So these 2 reactions first, sir. पहले हमको ना ये दोनों reaction याद करने होंगे. Succinyl CoA combines along with the glycine to form delta ALA. And delta ALA is getting converted to what, sir? Porphobilinogen. Then, now in the very first reaction, because delta ALA is forming there, the name of the enzyme will be ALA synthase. Okay. And this enzyme also requires a coenzyme that is your PLP. What is the PLP full form? Yesterday I taught you pyridoxal phosphate. And that pyridoxal phosphate is coming from vitamin B6. Okay. Now, and the second enzyme, second reaction, what is going to happen? There will be removal of water molecule from here. And whenever there is a removal, it is D. And water के लिए हम use करेंगे word dehydratase. Remember, it is not dehydrogenase. Dehydrogenase means removal of only hydrogen. There is oxidation reaction, but this is dehydratase. And the full name of this one will be the ALA dehydratase. So पहले आप आराम से first we are going to learn these two reactions now. Is it okay? So succinyl CoA combines along with the glycine to form delta ALA. Delta ALA will be getting converted to what's a porphobilinogen. Porphobilinogen. After that, this porphobilinogen will undergo like three reactions. Euro, copro, proto. Now, what is this Euro, copro, proto, sir? Europorphyrinogen. Copro porphyrinogen. Acha. And proto porphyrinogen. So you are able to see that the remaining name is absolutely the same. They said only you have to remember Euro or copro and proto. So europorphyrinogen that gets converted to coproporphyrinogen, that gets converted to what, sir? Protoporphyrinogen. Protoporphyrinogen. Now, my dear doctors, everyone knows very well in medicine, in our MBBS, wherever any word is ending with ogen, that means it is inactive. If it is ending with ogen, it is inactive. I'll give you one best example: trypsinogen, inactive. One very famous thing, no? Angiotensinogen. Angiotensinogen. That is nothing but, yes, getting activated to angiotensin and a pepsinogen. Yes, of course. So exactly the same thing happens here also. This will get activated to protoporphyrin. 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 So protoporphyrinogen is getting converted to protoporphyrin. अब जैसे ही यहाँ पर ये फॉर्म हो रहा है, आई होप ऑल ऑफ यू आर एबल टू रिकॉल. हेलो जी, ये प्रोटोपोरफायरिन कहीं सुना सुना सा लग रहा है. प्रोटोपोरफायरिन इज गेटिंग गोइंग टू गेट कन्वर्टेड टू हीम जस्ट बाय एडिंग व्हाट सर, आयरन. हो गया, दैट्स ऑल. अब आपको यहाँ पर लगाना होगा आयरन एंड दैट इज गोइंग टू बिकम व्हाट सर, योर हीम. And because you are going to add iron here in the form of Fe plus 2 ferrous, and therefore the name of the enzyme here will be ferrochelatase. Therefore, the name of the enzyme will be ferrochelatase. Done? So this is how I want my student to remember about the heme synthesis. Heme synthesis आपको ऐसे याद रखना है, आसानी से, such a simple thing. So first you are going to learn these two reactions here, sir. पहले आप ये दो reaction पढ़ोगे. And after that you learn about the Euro, copro, proto, and after that add the iron, sir. That's all that. Now let me add like the points that you have to learn here, sir. Okay. First of all, ALA synthase here, sir. This is the rate limiting enzyme of heme synthesis. This is the place where you are going to control heme synthesis, sir. That is the rate limiting enzyme. Number two, which was also asked in the FMG exam, sir. ALA dehydratase is inhibited by lead, plumbum, and then even ferrochelatase is also inhibited by lead, plumbum. They'll be giving that example now. A painter was painting and all those things. So paint में क्या होता है? Lead होता है, and that lead is going to inhibit these two substances. A person working in the paint industry, something related to paint, they will be giving sir. So whenever the person is painting, the paint में क्या होता है? Lead होता है, and that will be inhaled, and that is going to inhibit these two enzymes. और अगर ये दोनों enzyme inhibit हो जाएंगे तो heme form नहीं होगा sir. But basically in biochemistry here, I want to teach you that. Remember, lead is inhibiting which two enzymes, sir? One is ALA dehydratase, another one is ferrochelatase. That's all. Okay. By this, we are done with the synthesis of heme, sir. Now, once synthesis is done here, let us go ahead now. The last and final thing is degradation of heme. Degradation of heme, sir. Now, degradation of heme, my dear friends, do you want to learn this topic conceptually? Shall we learn this topic like conceptually, sir? Yes, from the scratch. Let us learn from the beginning. Why? Because yes, this topic is also very much important. Have you all heard about bilirubin? Conjugated bilirubin, unconjugated bilirubin and all. Yes, that is very much important because यहाँ पर क्या होता है ना, hyperbilirubinemia will lead to jaundice, and for that jaundice you have to have this you know clarity of this concept here, sir. तो चलिए हम इसको ना let us study from the scratch, and it's like a nice story here, sir. Nice kahani. I'll tell you slowly. Please remember that carefully here, sir. Remember, where will erythropoiesis take place? Erythropoiesis means formation of RBC. That will be taking place in our bones, the bone marrow. So in the red bone marrow, there will be formation of RBC, and formation of RBC is known as erythropoiesis. And after the formation of RBC, they will be transported out into the blood. And in the blood, RBC will be happily enjoying for 120 days. That is their lifespan. And after 120 days, that old RBC, the Buddha RBC, will go to its graveyard. And what is a graveyard of RBC, sir? Spleen. So the old RBC is actually going to the spleen, sir. And in the spleen, what will happen? The macrophages will eat the RBC, and then they are going to break down the RBC. Let's go break kar denge. And breakdown of RBC is known as what, sir? Hemolysis. अच्छा यार ये बताओ, hello, what is RBC filled with, sir? RBC के अंदर ना माइटोकॉन्ड्रिया होता है, न्यूक्लियस होता है, कुछ नहीं होता है. Inside the RBC will be actually having the hemoglobin only, sir. So once it is going to break down, so what is going to happen? Free hemoglobin will be released. Now again recall, in the hemoglobin, this globin will be actually the protein part, whereas heme will be actually the non-protein part, sir. Globin will be the protein part and heme will be the non-protein part. Up, let us deal with both of them one by one. Now this globin, which is protein part, will actually break down, and it is going to break down to form what, sir? Amino acid. Why? Because it's a protein, and protein is made up of amino acid. And what is the fate of this amino acid, sir? Amino acids will be reutilized in our body. Oh my God. We can't even think of excreting the amino acids, sir. Some students in some of the classes, they'll be answering, sir, ये end में form हो गया है, तो end product होगा, end product will be excreted out. No. Amino acids, just now I taught you, इसीलिए तो मैं पढ़ा रहा हूँ आपको. अभी मैंने आपको पढ़ाया, they're required for formation of many important substances. Melanin, melatonin, serotonin, पता नहीं क्या क्या, है ना? So for all that reasons, amino acids again reutilized in our body. Now what about heme, sir? Heme will actually undergo breakdown. It is going to break down to finally form bilirubin. Oh. So what is the end product of degradation of heme? Heme का degradation क्या होता है, sir? What is the end product of degradation of heme, my dear friends? So remember, that is actually bilirubin. ये बहुत important है, sir. Heme will break down to form bilirubin, sir. Now bilirubin का कुछ काम है क्या हमारी बॉडी में? सर कहानी समझ में आ रही है? स्टोरी. Is there any function of bilirubin inside our body? No, sir. There is no function. If there is no function, what we have to do? We have to actually excrete that out. You have to excrete that out. इसको भी excrete करना है. But the problem here is that bilirubin cannot be excreted out in urine. Why? Because it is insoluble. Bilirubin is insoluble. Understanding? ये वाटर में insoluble है, तो obviously क्या होगा? वो हमारे यूरिन में एक्सक्रीट नहीं हो पाता है. तो क्या किया जाए? What you have to do right now? We have to make it soluble. And to make it soluble, again comes your liver. बेचारा liver. देखो सर कितना काम कर रहा है liver, है ना? Liver works a lot. So now this bilirubin has to go from the spleen to the liver. From the spleen it has to go to the liver, sir. Okay. लेकिन ये कैसे जाएगा? It is going to go ahead with the help of a transporter in the blood. And who is the transporter in the blood, sir? That is albumin. I hope you all know albumin. Albumin is actually one of the plasma protein, and that albumin is going to form a complex with bilirubin. And albumin and the bilirubin together, they're going towards the liver. और ये क्या करेगा? यहाँ पर liver में bilirubin को drop कर देगा. And now once the bilirubin is dropped here, yes, we are actually going to do a conjugation reaction here, sir. Conjugation. We are going to combine this bilirubin with some substance here, conjugation. Yes, so we are going to combine this bilirubin with which substance here? That is UDP glucuronic acid. UDP glucuronic acid. I told you very well. Glucuronic acid is the one. अगर आप इसको UDP के साथ में अटैच कर दोगे, that will be active form of the glucuronic acids. ठीक है ना? UDP glucuronic acid. And now it is going to form what, sir? It's gonna become bilirubin diglucuronide. Two molecules of glucuronic acid will combine, sir. Bilirubin diglucuronide, sir. ठीक है ना? अब यार यहां से देखो टाइम पास इजी है ना. इन अदर सब्जेक्ट्स यू हैव टू मग अप दिस वन कि विच इज कॉन्जुगेटेड, विच इज अनकॉन्जुगेटेड, विच इज सॉल्युबल, विच इज इनसॉल्युबल. वेलकम टू बायोकेमिस्ट्री. इट्स वेरी इजी नाउ. Can I call this bilirubin diglucuronide as a conjugated bilirubin, sir? Bilirubin diglucuronide is a conjugated bilirubin, and conjugation we are going to do to make it soluble. Why we are doing conjugation? To make it soluble only. So definitely it is soluble. Now this bilirubin will be unconjugated bilirubin. Unconjugated will be actually insoluble bilirubin, sir. तभी तो हम conjugation कर रहे हैं. That is the reason why we are doing the conjugation there, sir. So normal bilirubin will be the unconjugated bilirubin, and that is insoluble. Whereas अगर आप conjugation आपने कर दिया, that is going to become conjugated bilirubin. Okay. अब क्या होगा? Now it is soluble. So from the liver, now what is going to happen? This bilirubin from the liver will actually go to, come on sir, gallbladder. And from the gallbladder, what is going to happen? Gallbladder में जाएगा और यहाँ से फिर वो क्या ना bile duct form होता है. And that bile duct will be finally going and draining the bile into the intestine, sir. Second part of the duodenum. We'll study about that in the anatomy part. Don't worry about that one. So now this conjugated bilirubin via bile duct will be actually going to the intestine. अब intestine में क्या होता है? In intestine, the bilirubin which is coming here, conjugated bilirubin, the soluble bilirubin, will be getting converted to number one, stercobilin, or else number two, urobilin. And this is converted with the help of the normal flora of the intestine, sir. Normal flora of the intestine, and that one you actually have to learn in the microbiology part. Okay, na microbiology mein padhoge aap. Now from the name itself, you are able to understand stercobilin will be excreted out in the stool, whereas urobilin will be excreted out in the urine. That's all, sir. Nice kahani. Kya baat. Are you understanding here? Beautiful story here. है ना? So I'm telling you the story right from the formation of RBC, sir. So RBC form होता है. After the lifespan is done, it is going to spleen. In the spleen, it is going to break down. Heme, from heme, we are getting bilirubin. But bilirubin is insoluble. To make it soluble, we are sending to liver. So please remember that very, very carefully, sir. When you're doing the conjugation, solubility बढ़ता है कि कम होता है? When you're doing the conjugation reaction, will the solubility increase or decrease, sir? Remember, solubility increases. We are doing conjugation to make it soluble. So once we are making it soluble here, it will go to intestine, convert to stercobilin, urobilin, excrete in the stool and the urine, sir. Crystal clear concept. Crystal clear concept, sir. And by this, we are done with this topic here, sir. So in hemoglobin topic, this is what we have to actually remember, my dear friends. The structure of hemoglobin, the structure of heme, then the synthesis of heme and the breakdown of heme, sir. We are done with this. Okay. So hemoglobin topic bhi ho gaya. And here we'll take a small break here, sir. यहाँ पर एक में एक छोटा सा break लेंगे. And after the break, we'll start with the lipids topic freshly, sir. ठीक है ना? Lipids topic हम पढ़ेंगे. Dr. Rekha, that is why actually, see, I wrote here. Dr. Rekha, I wrote here, this is taking place in intestine, right? ठीक है ना? Stercobilin will be going out in the stool. No problem with that one. But I told here urobilin will be going in the urine. अब यार ये तो intestine में है, तो intestine से urine में कहाँ जाएगा? कैसे जाएगा? Dr. Rekha, That urobilin I wrote here as going in urine here, but how it is going in the urine cell? Urine तो kidney से form होता है, तो वहां पर कैसे जाता है? तो वापस क्या होता है? Again, what is going to happen is from the intestine that urobilin will be actually absorbed back. It is absorbed back. So intestine, entero, liver, hepatic, enterohepatic, enterohepatic circulation कहते हैं उसको. तो फिर से वापस हो जाता है, फिर वहां से ब्लड में जाएगा. And from the blood, again it will go to kidney, in the kidney filtered, and then from there the urine will go in urine. अब ये intestine से urine में कहाँ जाएगा? कैसे जाएगा? It will be absorbed back. So that is why it is known as enterohepatic circulation कहते हैं. Gallbladder का क्या function है? It is helping in storage of bile. It is helping in concentrating the bile. तो बाइल जो है ना, बाइल में बहुत सारी चीजें होती हैं. बाइल will consist of bilirubin, it will have bile acids, bile salts. All that bile will go to gallbladder, and from the gallbladder it is stored and concentrated, and from there it is actually going to the cystic duct and then bile duct and then intestine. And gallbladder जो आपका सवाल था, it will be helping in storing the bile and concentrating the bile. ठीक है ना? चलिए. So by this topic is also very, very clear, sir. So we are now going to start with the lipids topic, sir. And in the lipids topic, the first thing that we are going to learn is the chemistry of the lipids, sir. Again, the same thing. First, we'll learn the basics, and then we'll actually learn about the lipid metabolism, sir. Chemistry of lipids. Okay. And as usual, every topic we're going to start from the scratch. And first of all, can anyone tell me what is the meaning of lipids, sir? Remember, lipids will be esters of fatty acid and glycerol. Lipids are esters of fatty acid and glycerol, sir. Esters of fatty acid and glycerol. Now, what is the meaning of this one? Esters of fatty acid and glycerol. You have to actually go back to the basics, sir. I think आपने organic chemistry पढ़ा होगा ना? In organic chemistry, there is a basic thing, sir. Whenever a carboxylic acid reacts with alcohol, finally it will be forming ester with the release of water molecules, sir. Whenever a carboxylic acid reacts with an alcohol, it is going to form what's an ester. This is known as esterification reaction. You all know very well a carboxylic acid will be actually having formula will be COOH. On alcohol, formula will be OH. Therefore, ester will be like C double bond O and O, sir. Ester will be COO. Fine. So now why I'm telling you about this one here right now. मैं आपको इसके बारे में अब क्यों बता रहा हूँ? क्योंकि welcome here, sir. Out of all the carboxylic acids that available here, many carboxylic acids are there. Out of them, we are selecting which one here, sir? Fatty acid. The fatty acid belongs to which group here? Carboxylic acid, sir. And out of all the alcohols which are available, sir, out of all the alcohols, we are selecting which alcohol right now? Glycerol. So whenever you react a carboxylic acid with the alcohol, we'll be getting what here? Ester. And this is the ester which is known as what, sir? Lipids. It's very simple. So what are lipids here? So lipids are esters of fatty acid as well as the glycerol. So if you are like, you don't want to remember the entire definition or something like that, at least remember like 2 words here, sir, from today onwards. The moment you listen the word as lipid in any subject, okay, now lipid is made up of 2 things here, sir. Number 1, fatty acid, another one is glycerol, sir. So one will be fatty acid, another one is the glycerol. Okay. Now, for example, if this is like one glycerol molecule here, you don't have to remember the entire structure of that one, sir. Just man lo ke this one will be the glycerol. And on one glycerol, how many fatty acids will be attached? Remember, there will be like 3 fatty acids attached. There will be 3 fatty acids attached on one glycerol molecule. So therefore, that is the reason why we'll call this one as what, sir? Tri. Why? Because 3 fatty acids are attached. And it is glycerol. So therefore, triglycerides. Triglycerides is the another name of lipids. And lipids are also known as what, sir? In general, as fats. Simple stuff. Okay. So because there are like 3 fatty acids attached on one glycerol molecule, so therefore it is known as what's a triglyceride. Triglycerides are also known as the lipids, also known as the, in simple English, we'll call them as fats here. So, okay, definition is very clear. Now, my dear friends, remember, It means that what is a lipid made up of, sir? Lipid is made up of two things there. That is your glycerol and the fatty acid. But we are not going to learn anything about the glycerol in this topic right now, sir. Glycerol is an alcohol, and alcohol metabolism will be a separate metabolism. We're not going to worry about that one. Now, this topic is all about what here, sir? Fatty acids right now, guys. Understanding your point here? So simple. So before the break, we actually studied about what here? Before the break, we studied about the proteins topic. Proteins topic is all about what's an amino acid. And now we are going to learn about the lipids topic. Lipids topic is all about what's a fatty acids. Understanding? So the only thing we have to concentrate right now is on fatty acids. So again, let's start from the scratch. Come on. What is a fatty acid, sir? Okay, now, what is a fatty acid? Just now I told you that fatty acid belongs to the category of carboxylic acid. So the functional group will be COH. And to this carboxylic acid, you have to attach a long chain of carbon, sir. There will be a long chain of carbon attached, almost like 18 carbon, 20 carbons will be attached there. Okay, now, बस इसी को आपको इंग्लिश में लिखना है. What is a fatty acid? Fatty acid will be long chain carboxylic acid, sir. Fatty acids will be long chain carboxylic acid, guys. ठीक है ना? So there will be a long chain of carbon attached to carboxylic acid. That's also, that is the definition. It's so simple. That itself is the definition. I don't know why students don't study. So fatty acid बड़ा आसान है. There will be a long chain of carbon attached to carboxylic acid, sir. ठीक है ना? Next one. Once you are done with the definition, now the next thing will be the classification. And the first classification of the fatty acids will be based on number of double bonds. And where we have to look for this double bonds here, sir? We have to look for this double bonds in the long chain of carbon which is attached here, sir. In this long chain of carbon. Because we know that carbon has the ability to form the multiple bonds. So there will be carbon-carbon single bond, carbon-carbon double bond, as well as carbon-carbon triple bonds will also be there. I hope you all remember, I think your chemistry is recalled right now, sir. We learned about that alkane, alkyne in our lower classes. So we know about that. Okay. So now based on double bond, the fatty acids are actually divided into two categories here, sir. Number one, there will be saturated fatty acid. Saturated fatty acid. And number two, there will be unsaturated fatty acid, sir. Now, what is the meaning of saturated and unsaturated fatty acid? Remember, saturated का मतलब क्या होता है? There will be no double bond in that one. Unsaturated means there will be a double bond. And these unsaturated fatty acids are in turn divided into two categories here. One will be MUFA, another one will be the PUFA, sir. Now, what is the meaning of this MUFA, PUFA? Mono unsaturated fatty acid and poly unsaturated fatty acid. Mono means it will consist of only one double bond, and if there is more than one double bond, it will be known as poly unsaturated fatty acids. All simple, sir. Very easy topic, sir. Lipids. ठीक है ना? Are you all done? Yes, thank you. Now, first classification completed here. Now, the second classification will be again the same thing based on nutritional requirement. Based on nutritional requirements. Now, based on nutritional requirement, it is again divided into the same things, essential and non-essential fatty acid. And we have to just remember the essential fatty acid. And my students are already expert in the definition. What is the meaning of essential fatty acid? Are they produced in our body? Come on, my dear friends, answer. Essential, are they produced in our body? No. They are not produced in our body, and they have to be actually, taken in the, essentially taken in the diet, essential fatty acid. Now, essential fatty acids will be actually three of them. Number one, they will be linoleic acid. Number two, there will be linolenic acid. And number three, you'll be having the arachidonic acid, sir. Arachidonic acid. So these three will be the essential fatty acid. And any other fatty acid that you come across in your life, all of them will be the non-essential, sir. So that is the reason why we actually don't have to learn about the non-essential fatty acid. Just learn about the essential fatty acid. The essential, there'll be three of them: linoleic, linolenic, and arachidonic acid, sir. अब यार इसमें ना पढ़ना क्या होता है? What you have to learn here? The nomenclature. Remember, the nomenclature of linoleic acid will be 18:2, 9:12. So 18:2, 9:12. Now, what do you understand? What do you have to understand from these numbers here? Remember, 18 represents the number of carbons. So linoleic acid is having how many carbons, sir? 18 carbons. I told you right just now, the carbon, there will be a long chain of carbon. And then the next number is representing the number of double bonds. So how many double bonds do you have? 2. And when you know that there are 2 double bonds, then you have to know the position of the double bonds, sir. So one of the double bond is located on 9 carbon and another double bond will be located on 12 carbon, sir. It's so simple. So that is how you have to understand the nomenclature, sir. 18:2, 9:12, sir. Apart from that, there is one more nomenclature. I think you people actually have heard about that omega-3 fatty acid and omega-6 fatty acid and all. So my dear friends, how to remember this one? That is so simple. First number minus the last number, sir. That's all. So 18 minus 12 will be 6. So this will be omega-6 fatty acids, sir. So follow my method here. The life will become easy. Chalo. Let us do the same thing for the other ones also here, sir. Now, what is the nomenclature for linolenic acid? It will be 18:3, then 9, 12, 15. So tell me right now, sir, how many number of carbons do we have here? 18 carbons. And how many double bonds do we have here? 3 double bonds. Where are they located? On the 9 carbon and the 12 carbon and the 15 carbon. So 9, 12, and 15 carbon. And first number minus the last number. Yes, very nice. Dr. Vandana, Dr. Vishal, Dr. Manjur Islam. Yes. 18 minus 15 will be 3. So this is omega-3 fatty acid. Similarly, arachidonic acid will be actually 20:4. And then it will be 5, 8, 11, and then 14, sir. So 20 carbon, how many double bonds? 4 double bonds. They're located on 5th one, 8th one, 11th, and 12th, and 14 carbon. So 20 minus 14 will be 6, and this will be the omega-6 fatty acids. This is what you actually have to remember about this classification. Essential fatty acids में आपको याद रखना होगा sir. ठीक है ना? So there are 3 essential fatty acids, and all the 3 essential fatty acids, you have to remember their nomenclature, sir. Next one. Now all these 3 are essential fatty acids. Out of this one, which is the most essential fatty acid, sir? So remember, the most essential will be the linoleic acid. The most essential will be the linoleic acid, sir. Done? That's all we are done. That is all that you have to remember about the fatty acids, sir. Definition done and the classification done, sir. So one classification is based on number of double bonds, saturated, unsaturated. Another classification is based on nutritional requirement. Linoleic acid, linolenic acid, and arachidonic acid. These 3 are essential, remaining are non-essential, sir. Done. So by this topic is completed here, sir. Now let us go to the next topic. After learning about fatty acid, now please write on the heading as classification of lipids. Okay. And I want everyone to be alert here in the classification of lipids. I'll tell you about like 6 diseases here, and at least one will be coming from out of the 6 diseases. At least one will be coming in the exam, and you have to remember that one, sir. Okay, na? So चलिए start करते हैं. First of all, lipids are actually divided into 2 categories here, sir. Simple lipids and the complex lipids, sir. Now, what is the meaning of this one? Simple lipid means they will just have fatty acid along with alcohol. That's all. But complex lipid will have fatty acid along with alcohol, along with some third substance. Simple lipids will just have fatty acid along with alcohol, whereas complex lipids will be having fatty acid along with alcohol, along with some third substance. Okay. Now, in the simple lipids, first of all, let us deal with the simple lipids. They're again divided into 2 categories. In one of the category, you'll be having fats or oils. In another category, you'll be having the waxes. Now, what is the difference between these 2? Fatty acid along with alcohol, and the alcohol will be glycerol. So if the alcohol is glycerol, then it is simple one, sir. That is your fats or oils. Along with the fatty acid, if it is any other alcohol except glycerol, any other alcohol except glycerol, then it will be the it will be the waxes. Okay. Acha, that means in my table you can understand very, very clearly here, my dear friends. Okay, simple lipids are in turn divided based on alcohol. If the alcohol is glycerol, then it is fats or oils. If the alcohol is any other alcohol, then it will be waxes. Okay, next one. Now moving on to the other side, sir, let us deal with the complex lipids right now. Fatty acid plus alcohol, I can call it as lipid. Along with this lipid, sometimes the third substance will be a phosphate group. If there is a phosphate group, then phosphate plus lipid is known as what, sir? Phospholipid. Okay. Sometimes there will be a third substance that is carbohydrate. If there is a carbohydrate, we'll be using the word as glycolipids. Or sometimes there will be actually the protein. And lipid along with the protein is known as what, sir? Lipoprotein. Are you understanding this one? So sometimes there'll be a phosphate group. So therefore it is known as phospholipid. If it is carbohydrate, glycolipid. And suppose if there's a protein present along with the lipid, so it is known as lipoprotein. I think you people have all heard about that one, no? VLDL, LDL, HDL and all. You know? What is that one? Low density lipoprotein. High density lipoprotein, something like that. So these are all like your lipoproteins. They come under this category here. And now, don't think that this topic is done here, sir. Hello. And now we have to learn about all the three of them one by one. That is your phospholipid, glycolipid, and the lipoprotein. So first, let's get started with the phospholipid, sir. Please write this in the center of the page here. Phospholipid. What is a phospholipid made up of? So it is made up of fatty acid along with alcohol, along with the phosphate group. Okay. That is in turn divided into two categories here, sir. That is fatty acid along with alcohol and phosphate group. Fatty acid along with alcohol and the phosphate group, sir. Okay. That means the moment you look at my table, you can easily understand that, oh, they're again divided based on what? They're again divided based on alcohol. So if you, my dear friends, if you look at this topic in your textbooks, you'll actually go mad, sir. In the textbooks, it is given in a very complicated manner. I'm giving you a simple table here so that just before the exam, you can easily revise this one here, sir. है ना? तो जैसे आप टेबल देखोगे यहाँ पर क्लियरली समझ में आ जाएगा कि it is divided based on what, sir? Alcohol. Sometimes the alcohol will be glycerol. So therefore, what is the name of this one? Glycerophospholipid. Okay. Sometimes the alcohol will be sphingosine. I don't know whether you have heard about this one or not. Yes, my dear friends, sphingosine, alcohol. So therefore, if the alcohol is sphingosine, then it is known as what, guys? It is known as sphingophospholipid. Yeah, it's so simple. So phospholipid is again divided into two categories here. That is, one is glycerophospholipid, another one is sphingophospholipid, and that is actually based on what's alcohol. Agar alcohol glycerol hoga, glycerophospholipid. If the alcohol is sphingosine, sphingophospholipid. Now, in this sphingophospholipid, one of the example here will be the sphingomyelin. And sphingomyelin is metabolized by sphingomyelinase. Sphingomyelin is metabolized by sphingomyelinase. You just have to add A's there. Now, in some unfortunate people, this enzyme will be deficient, or agar ye deficient hoga, it will lead to a disease known as Niemann-Pick disease. What is the name of the disease, sir? Niemann-Pick disease. So your focus should be actually here. What is the name of the disease? Niemann-Pick disease. And what is the enzyme deficient in this one, sir? That is sphingomyelinase. Done and done and done, sir. This is what you have to remember about the phospholipids, sir. So phospholipids are actually divided into two categories, glycero and sphingophospholipid. A sphingophospholipid, one of the substance will be sphingomyelin, and sphingomyelin is metabolized by sphingomyelinase. अगर ये enzyme deficient होगा, so it will lead towards a Niemann-Pick disease. So what is the name of the disease? Niemann-Pick disease. And what is the enzyme deficient disease? The enzyme deficient will be sphingomyelinase. कोई दिक्कत? Hello, my dear friends. Any problem here? Niyato problem. Now we are done with the phospholipids. Now let us start with the glycolipid, the second one. Write down again in the same manner, glycolipid in the center of the page. Fatty acid plus alcohol plus carbohydrates. Fatty acid plus alcohol plus carbohydrates. ठीक है ना? Now these glycolipids are again divided into two categories here, sir. There will be fatty acid along with alcohol along with carbohydrate. There will be fatty acid along with alcohol and along with the carbohydrates. Okay. Now sometimes the carbohydrate will be monosaccharide. Sometimes the carbohydrate will be oligosaccharide. You know, not this one, like monosaccharide will be having like only one single unit. And then oligosaccharide means few, poly means many. ये तो simple words है आपको पता है sir. Mono means one, di means two, tri means three, oligo means few, and poly means many. Achha, by the way, what is the name of this one? If there is a monosaccharide, then it is known as cerebrosides. And if there is a oligosaccharide, then it is known as gangliosides. First, slowly and neatly you learn about the names, sir. So I repeat again, the glycolipids are in turn divided into two categories there, sir. That is your cerebrosides as well as the ganglioside. Now, out of these two, first let us deal with the cerebrosides, sir. Cerebrosides are in turn divided into two categories right now, sir. Fatty acid plus alcohol plus monosaccharide. Fatty acid plus alcohol plus monosaccharide. Sometimes the monosaccharide will be glucose and sometimes the monosaccharide will be galactose. कुछ याद आ रहा है आपको? Are you able to recall? Yesterday we have learned about the galactosemia in detail. In that I told you UDP galactose, the activated form. And UDP galactose is required for formation of number 1, keratin sulfate, number 2, myelin, and number 3, I told you glycolipid. Glycolipid form करने के लिए galactose जरूरी है sir. Galactose is important, sir. Understanding? So I told you 3 things. You can turn the page back and see. I repeat again. UDP galactose is required for the formation of keratin sulfate and then myelin as well as the glycolipids. ठीक है ना? Now, if the monosaccharide is glucose here, then it is known as glucocerebroside. And if it is galactose here, it is known as galactocerebroside. Glucocerebroside will be metabolized by an enzyme. The name of the enzyme will be glucocerebrosidase. आपको सिर्फ A's लगाना होगा सर. Glucocerebrosidase. Sometimes this enzyme will be deficient. Deficiency of this enzyme will lead to a disease known as Gaucher's disease, sir. This is Gaucher's disease. So in biochemistry, you have to remember the name of the disease and the enzyme deficient, guys. So in Gaucher's disease, what is the enzyme deficient? That is glucocerebrosidase. Very good. Crumpled tissue paper appearance. Good. After that, galactocerebroside will be actually metabolized by galactocerebrosidase. आपको सिर्फ A's लगाना होगा. But here the problem is that this enzyme will be in two forms, sir. One is alpha, another one is the beta, sir. One is alpha galactocerebrosidase, another one is beta. Now, If alpha galactocerebrosidase is deficient, it will lead to Fabry's disease. Okay. And if beta is deficient, it will lead to Krabbe's disease. So you can just remember with the rhyming word there. Alpha, remember Fabry's, and beta, you remember Krabbe's. Krabbe's. ठीक है ना? Krabbe's. Krabbe's, you remember like beta. ठीक है ना? So alpha galactocerebrosidase deficiency will lead to Fabry's disease, and beta galactocerebrosidase deficiency will lead to Krabbe's disease. Sir. Done. हो गया. This is what you have to remember here. Now, please look at this classification again, sir. I told you glycolipids are actually divided into two categories, na, sir. That is your cerebroside and the gangliosides. है ना? So now we are actually done with what here? Cerebroside completed here, sir. ये हो गया. And now let us go towards the other side, that is ganglioside, sir. So I'm going to write down this separately here below. Now, gangliosides, nothing much, the classification here, sir. They are undergoing metabolism, but the problem here is the name of the enzyme will be different. That is hexosaminidase. One is alpha hexosaminidase, another one will be the beta hexosaminidase. So the gangliosides are actually metabolized by which enzyme? Hexosaminidase. One will be alpha, another one is beta. If alpha hexosaminidase will be deficient, then it will lead to Tay-Sachs disease. If the beta hexosaminidase is deficient, then it will lead to what, sir? The Sandhoff's disease, sir. Okay. So deficiency of alpha hexosaminidase will lead to Tay-Sachs disease. Deficiency of beta hexosaminidase will lead to Sandhoff disease. That's all. So जैसे मैंने आपको कहा था, as I told you, how many diseases, sir? 6 diseases. Number 1, Sandhoff. Number 2, Tay-Sachs. Number 3, Krabbe's. Number 4, Fabry's. Number 5, Gaucher's. And then number 6 will be the Niemann-Pick disease, sir. That's all. We are done with this one here, sir. So in your FMG exam, they have asked this a lot of times, sir. Gaucher's disease पूछा जाता है, and then Tay-Sachs and all this will be asked here, sir. Now let us go back to the main classification. Do you still remember? We are done with glycolipids. We are done with the phospholipids here. Now please look at the main classification here. The last thing which is left out here is actually the lipoprotein. But lipoproteins, we are not going to learn it right now, sir. In this topic here, we are going to learn them in the lipid metabolism, sir. Lipid metabolism, sir. Clear? And by this, we are actually done with the chemistry of lipids here, sir. Chemistry of lipids completed here, guys. Okay. So this is what you have to remember, the chemistry of lipids here. Now, once this topic is done here, the next topic that we are going to begin right now will be the lipid metabolism. इसीलिए मैं आपको ना बार बार कह रहा था, that is why I'm telling you that this topic is very much easier, sir. That is why I've given you a nice break so that now you can learn it perfectly, sir. Okay. Shall we go ahead now? Now, first, what you do is the planning here, sir. The planning here is that lipid metabolism topic, we are going to divide into 3 categories here. Number 1 category, we are going to learn about the cholesterol. We are going to learn about the synthesis of cholesterol and breakdown of cholesterol. Number 2 category, we are going to learn about the fatty acid. Synthesis of fatty acid and breakdown of the fatty acid. And number 3 category, we are going to learn about the lipoproteins, sir. Khatam. So first we are going to learn about the cholesterol, and then we are going to learn about fatty acid, and then finally we are going to learn about what's a lipoprotein. If you learn these 3 topics here, our lipid metabolism topic will be like done, done, and done, sir. Easy. So now first let me deal with the cholesterol right now, sir. So as I told you, first we are going to learn about the synthesis. Synthesis of cholesterol. Synthesis of cholesterol begins from where? Remember, it starts from acetyl-CoA. Acetyl-CoA will combine with another molecule of acetyl-CoA, and these two will combine together to form aceto-acetyl-CoA. Wow. इससे आसान और क्या चाहिए जिंदगी में, सर? Such an easy thing. Acetyl-CoA, acetyl-CoA, aceto-acetyl-CoA. Now, in this acetyl-CoA, add one more molecule of acetyl-CoA. It is going to become HMG-CoA. HMG-CoA, sir. Now this HMG-CoA will get converted to mevalonate. And this mevalonate will get converted to isoprenoid units. Isoprenoid unit will get converted to squalene. And finally, the squalene is the one which is going to form what's a cholesterol. That's all. HMG-CoA to mevalonate, mevalonate to isoprenoid unit, and isoprenoid unit to squalene, squalene to cholesterol. Dr. Akhil is telling it was asked this time, sir. Okay. इस बार ही पूछा गया था. Now, my dear doctors, do one thing here. Take another color pen or pencil and write down there. No need to learn this pathway, sir. ये entire pathway आपको पढ़ने का जरूरत नहीं है. Then why I am teaching you this pathway here, sir? Only and only for one enzyme, and that enzyme is HMG-CoA reductase. And you cannot forget this enzyme lifetime, lifelong. आप इसको नहीं भूल सकते हो. क्यों? I'll tell you, sir. Why? I'll tell you. HMG-CoA reductase, this is actually the rate limiting enzyme. It is the rate limiting enzyme of cholesterol synthesis. Number one. HMG-CoA reductase is the rate limiting enzyme, sir. Number two, we all know the kind of life we are leading right now, sir. Like stationary life, sitting at one place for long classes. Onto that, like all junk food. Onto that, no physical activity, nothing. Okay, na? तो क्या होता है? In our bodies, hypercholesterolemia will be there, sir. Cholesterol level will be high. तो इसके लिए हम क्या करते हैं? In pharmacology, you will be having statin group of drugs, sir. And if you have learned pharmacology, I hope you know that. What is the mechanism of action of statins? Statins are going to inhibit HMG-CoA reductase, sir. Mechanism of action. So number two, this is the second point here. And yes, of course, what is the starting substance of cholesterol synthesis? Sir? Acetyl-CoA. That's all. I'm done with the cholesterol synthesis. This is what you have to learn. I'm telling you very, very clearly. Please don't waste your time in learning the entire pathway. No need to learn the entire pathway, sir. Only what you have to do is, whatever points I'm telling you, you learn those points only, sir. Or else I'll tell you one method here, sir. Remember, in this topic, I have to just learn how many points here, sir? 3 points, sir. In cholesterol synthesis, how many points you have to learn? 3 points. Number one, what is the rate limiting enzyme? HMG-CoA reductase. Number one. Number two, statins are going to inhibit HMG-CoA reductase. Number two. And number three, what is the starting substance of cholesterol synthesis? Acetyl-CoA. That's all. That's all, my dear friends. If you learn these 3 points, that is more than enough for your exam, sir. Cholesterol synthesis, ho gaya, done. So after my class, when you're going to revise this one, your focus should be only on these 3 points, sir. Make your life easy. Now, after the synthesis of cholesterol, my dear friends, heading लिखो degradation of cholesterol, sir. Come on, my dear doctors. Degradation of cholesterol. अब यार degradation of cholesterol में क्या होगा? Again the same thing. You just have to learn like 3 points here, sir. कैसे? How? Look here. When this cholesterol will undergo degradation, it is going to form 3 substances, sir. Number one, cholesterol is going to form all the steroid hormones in our body. This was asked in your previous exams, FMG में. All the steroid hormones in our body are formed from where only, sir? Cholesterol only. Don't even think about that one. Any steroid hormone, glucocorticoid, mineralocorticoid, testosterone, estrogen. समझ में आ रहा है, sir? Are you understanding me? हाँ. Hello, my dear boys. Are you understanding? Cholesterol is good or bad? Very good. It is the one which is going to form the steroid hormones. Number two, cholesterol is the one which is also going to form vitamin D. Vitamin D is also formed from the cholesterol only. And number three, cholesterol is the one which is also going to form your bile acids. So cholesterol is going to form steroid hormones. Number two, it is going to form vitamin D. And number three, it is also required for the formation of bile acids. That's all. So you just have to remember these 3 points here and you are done with the degradation of cholesterol. That's all. So my dear doctors, you have to remember like 3 points in degradation of cholesterol and you have to remember like 3 points in synthesis of cholesterol. Cholesterol topic completed here, sir. ठीक है ना? Now, my dear friends, welcome back here. हमारा planning क्या था, sir? What is our planning in lipids topic? Yes. In lipids topic, we are done with the first one. Cholesterol हो गया. And now, again, the same thing. We have to learn about the fatty acid. And in fatty acid also, you have to actually learn two things: synthesis of fatty acid and degradation of fatty acid, sir. Synthesis and degradation. बस दो ही चीजें पढ़ना है अपने को. पहले फॉर्म करेंगे, फिर ब्रेक करेंगे, सर. वही दुनिया का पुराना उसूल, पहले जोड़ो फिर तोड़ो. चलिए सर आगे बढ़ते हैं. नाउ फर्स्ट ऑफ ऑल वी आर गोइंग टू लर्न अबाउट द फैटी एसिड सिंथेसिस राइट नाउ, सर. प्लीज राइट ऑन द हेडिंग देयर एज फैटी एसिड सिंथेसिस. ठीक है ना. फर्स्ट रिमेंबर, फैटी एसिड सिंथेसिस विल टेक प्लेस इन साइटोसॉल. लोकेशन जरूरी है. वी हैव टू रिमेंबर द लोकेशन हियर, सर. आफ्टर दैट, if you people are like really following my class, sir, I think you are like attentive in my class. Hardly I'm taking the class for like few hours daily, not even like full day also. If you're attentive in my class, I told you yesterday only when I was teaching you about the, you know, glycogen storage diseases. Fatty acid कहाँ से form होता है? Fatty acid is formed from where? You remember? Acetyl CoA. Pyruvate to lactate, pyruvate to acetyl-CoA. And I told you when acetyl-CoA will be excess, that will lead to the formation of fatty acid and cholesterol. And when acetyl-CoA is excess, it is leading to the formation of fatty acid and cholesterol. So hyperlipidemia and hypercholesterolemia हमने लिखा था type 1 glycogen storage disease में. तो वहां से याद करो ना. Please recall from there ना. Very good Najib, very good Alisha, very good Akhil, very nice. हाँ जी. तो remember, फैटी एसिड कहां से फॉर्म होता है? फ्रॉम वेयर इज द फैटी एसिड फॉर्मिंग? असिटाइल कोए. So therefore, what is the starting substance here also? Acetyl CoA. Here also the starting substance is acetyl CoA. Acetyl CoA is first going to form malonyl CoA. अच्छा जी. And malonyl CoA is the one which is going to form your fatty acid. Topic khalas. Topic khalas. Done. So acetyl CoA will form malonyl CoA, and malonyl CoA is going to form what's a fatty acid. Now, how is this acetyl CoA going to form malonyl CoA? By addition of carbon in the form of carbon dioxide. And now, though my students are expert, the name of the enzyme will be carboxylase, and the full name has to be acetyl CoA carboxylase. साथ में substrate का नाम लगा दो, acetyl CoA carboxylase. And this is the third and the final enzyme requiring biotin as a coenzyme. Oh my God. Are you making the list? Are you making the list somewhere, sir? So acetyl CoA is going to form the malonyl CoA by carboxylation, and that is why the name of the enzyme is acetyl CoA carboxylase. Take it. And then the coenzyme required will be biotin, sir. Acha, because the fatty acid is forming here, therefore, what is the name of this enzyme here? It is fatty acid synthase complex. So that's all. ये तो और भी आसान हो गया. Okay. So therefore, my dear friends, forget about this one. Fatty acid is forming. Therefore, the name of the enzyme is fatty acid synthase only. So please concentrate on only and only this reaction. Your life will become easy right now, sir. Fatty acid synthesis हो गया. If I will be at your place writing FMG exam, अगर मैं FMG exam लिख रहा होता, तो in this topic, I just have to learn like 4 points. After class, we're going to learn only 4 points. Where is fatty acid synthesis taking place? Cytosol. Okay. What will be the rate limiting enzyme? Of course, the rate limiting enzyme will be acetyl CoA carboxylase. Second point. Acetyl CoA carboxylase requires which coenzyme? Biotin. And what is the starting substance? Acetyl CoA. वो तो आपको already पता है. That is a building block of what's fatty acids. So at least you make some concept and remember. Like proteins, आपको ना बचपन से पता है. You know that from childhood. Proteins are made up of amino acid. Similarly, in the lipids topic, remember fatty acids are made up of acetyl CoA. So acetyl CoA is like building block of fatty acids. Okay, so that's all. हम ये 4 point पढ़ेंगे और ये topic class. एक बार हम दोबारा repeat करते हैं. Let us try to learn in the class itself, sir. Fatty acid synthesis, we have to learn like 4 points here. Number 1, it is taking place in cytosol. Number 2, what is the rate limiting enzyme? Acetyl CoA carboxylase. And that requires which coenzyme, sir? Biotin, vitamin B7. And then finally, what is the starting substance? That is acetyl-CoA. Done, done, done. अब क्या करना होगा? We have to learn the degradation of fatty acids. बस time pass है sir biochemistry. Please write on the heading there as degradation of fatty acid. चलिए इसको break करते हैं. And now this topic is a little bit bigger topic, sir. ये थोड़ा बहुत ना एक it will take some 10 minutes of time, 10-15 minutes of time. तो थोड़ा सा attention here. And because it is a bigger topic, यार overview तो बनता है. अरे यार ये बताना overview help करता है कि नहीं करता है? Now, hello my dear doctors. Fatty acid is undergoing degradation. So first of all, you people tell me here, okay, fatty acid is forming from where, sir? So my dear friends, can you please tell me, fatty acid is forming from where? Fatty acid is forming from acetyl-CoA. Acetyl-CoA is the one which is going to form the fatty acid. अगर ये breakdown होगा तो वापस क्या मिलेगा? Fatty acid. Sorry, acetyl-CoA only. है ना? It's same thing. Amino acid is going to form protein. Protein will break down to form what? Amino acid. Similarly here, acetyl-CoA is going to form fatty acid. Fatty acid will break down to form what only? Acetyl-CoA only. Welcome. Welcome to my class. Fatty acid will break down to first of all form acetyl-CoA. Very easy. What is the name of this pathway? This is known as beta oxidation of fatty acid. So beta oxidation of fatty acid, what is the starting substance? Fatty acid. And what is the end product? Acetyl-CoA. And this acetyl-CoA will be further utilized for the formation of ketone bodies. Achha. I think you are able to recall our yesterday's class completely now. Type 1 glycogen storage disease में हमने लिखा था ketosis भी होता है, ketone body form होता है from the acetyl-CoA. You know. So because the ketone bodies are forming here, this condition or this pathway is known as ketogenesis. This pathway is known as what's the ketogenesis? Oh my God. That means in the degradation of fatty acid, how many pathways do we have to learn right now, sir? We have to learn like 2 pathways here. That's all. Even this topic will be done in 10-15 minutes, sir. Is it okay? Degradation of fatty acid, sir. Okay. The fatty acid will break down to form acetyl-CoA, and then acetyl-CoA is going to form what, sir? The ketone bodies. Guys. Only 2 pathways you have to learn. Out of these 2 also, beta oxidation is very much important here, sir. We have to learn that in detail. And after that, ketogenesis, it will be done in 2 minutes, sir. ये 2 minutes में हो जाएगा. Okay. Now, first of all, beta oxidation of fatty acid will take place in 3 steps. Number 1, we have to activate the fatty acid, sir. Activation of fatty acid. Activation of the fatty acid. And activation of fatty acid will take place where, sir? In cytosol. దాని తర్వాత ఏం చేయాలి? Transport చేయాలి. We have to transport this from cytoplasm into mitochondria. And there actual beta oxidation will take place. And that is known as beta oxidation proper. And beta oxidation proper will take place in the mitochondria. Understanding? Now, after writing this one, my dear friends, please put this in a box here. So this is like our overview right now. एक बार repeat करते हैं. So let us repeat this one without any confusion. It is worth repeating once. Degradation of fatty acid. Fatty acid will undergo degradation to form what's acetyl-CoA. And acetyl-CoA is the one which is finally going to form what, guys? Ketone bonds. Okay, now degradation of the fatty acid, it is known as what? Beta oxidation of fatty acid. It is going to take place in 3 steps. Activation, transport, and beta oxidation proper. And then after that, there'll be ketogenesis, guys. Done. I hope I'm not confusing you. I hope I'm not confusing you, my dear friends. Is it clear till here? Chalo. अगर ये clear है, तो let's get started with the first topic right now. So the first topic that we are going to begin right now is beta oxidation of fatty acid. Beta oxidation of fatty acid will actually take place in how many steps, sir? 3 steps. And the very first stage will be activation. Activation of fatty acid. And my student knows very well, activation kahan ho raha hai, sir? Cytosol mein ho raha hai. It is taking place in the cytoplasm. Now, activation is so easy that the fatty acid is activated to acyl-CoA. Fatty acid is activated to acyl-CoA. Please write down the spelling very carefully there. You know, don't think that Azam sir is writing wrongly here. Acetyl, मैं acetyl लिखूंगी. No, no, no. It is not acetyl. It is actually acyl. The fatty acid is activated to acyl-CoA, sir. Now I'll tell you what are the difference between these two right now. I mean, दोनों में फर्क क्या है, sir? What is the difference? So, my dear doctors, remember, what is the definition of fatty acid? What is the definition of the fatty acid, sir? Fatty acid will be actually having the long chain of carbon attached to carboxylic acid here. Attached to carboxylic acid. And now this fatty acid will be actually activated to acyl-CoA. तो मतलब क्या हो रहा है? The long chain of carbons will remain as it is. Just in the place of carboxylic acid, it is replaced by CoA, sir. Okay. That is how it is activated. हो गया, done. Now, can you please recall and tell me, from last 2 days we have been learning, how many number of carbons will be there in acetyl-CoA? Acetyl-CoA में कितने कार्बन होते हैं? How many number of carbons do you have in acetyl-CoA? 2 carbon. Exactly 2 carbon. है ना? Pyruvate will have 3 carbon. Link reaction. Pyruvate to acetyl-CoA. Acetyl-CoA will have like 2 carbons. Is it okay? So pyruvate to acetyl-CoA will exactly have 2 carbon. But acyl-CoA will have how many carbons, sir? Acyl means many carbon. That is the difference, sir. Okay, na? So my dear friends, please remember the difference here. In biochemistry, whenever you're going to come across acetyl and acyl, don't think they are same, sir. Okay, na? Acetyl means exactly 2 carbon, whereas acyl means many carbon. And you can clearly see they're like many carbons. Simple, ho gaya. Now, because acyl-CoA is forming here, therefore the name of the enzyme will be what here, sir? Acyl-CoA synthase. Name of the enzyme is very simple, and you know very well, whenever we activate any substance, we have to provide the energy, sir. And here energy will be provided in the form of ATP. That to how many? 2 ATPs will be given here. है ना? The boys will completely agree with me here. जब कभी भी हमको किसी को भी एक्टिवेट करना होता है तो एनर्जी देनी पड़ती है कि एनर्जी मिलती है? एनर्जी देनी पड़ती है कि मिलती है, सर? रिमेंबर, एनर्जी देनी पड़ती है. So to activate, we have to give the energy, and yes, here energy will be given in the form of ATP. Is it okay? Come on, my dear friends, is it okay? Now, beta oxidation of fatty acid will take place in 3 steps here, and we are done with the first step. Activation ho gaya. Fatty acid is activated towards acyl-CoA. Now, the second step. And the second step is transport. Now we have to transport that acyl-CoA into the mitochondria. Into mitochondria, we have to transport that concept. Okay, and now, welcome to time pass. I mean to say, welcome to biochemistry. मान लो कि this is the outer membrane of the mitochondria and this is the inner membrane of the mitochondria. Inside the mitochondria, this is the matrix. Outside the mitochondria, this one is the cytosol. And right now in the cytosol, we are having a substance known as acyl-CoA. Platform ready. Thank you, Dr. Sheikh the Brace. Okay, na, sir? Platform is ready. If you people have drawn this diagram till here, I request all of you to please stop writing now, sir. Please likhna band karo. Don't write with me. First look at the screen here. After that, I'll give you time for writing, sir. मैं आपको लिखने के लिए समय दूंगा. ठीक है ना? In cytoplasm, there is a substance known as carnitine. What is the substance, sir? Carnitine. Now this carnitine, okay, now that is our car. It is going to combine with acyl CoA. Kaise? How? It is going to combine with the acyl part. Acyl carnitine. Okay. And what is left out? CoA. Now this acyl carnitine will happily go inside the mitochondria. Please don't write with me. And acyl carnitine will be going inside the mitochondria and combining with, again, coenzyme A. Remember, the coenzyme A in the cytoplasm is separate and the coenzyme A in the mitochondria is separate. So it cannot cross the membrane. Separate pool here. Now this acyl part will react with the CoA to form what, sir? Acyl CoA. And what is remaining? Again, carnitine. And this carnitine will happily again come out here, sir. So, mera time pass. So therefore, what is going to happen now? The acyl CoA, which was in the cytoplasm here, sir, and now that acyl CoA is actually inside the mitochondrial matrix, guys. Kya baat hai, sir. Transport ho gaya. Okay. Now the students will be looking at this such a big diagram and they'll be confusing. Sala kahan se shuru kare, kahan khatam kare? It's so simple here, sir. So simple. Achha, by the way, what is the name of the enzyme? Outside, there'll be one cat here, sir. That is known as cat1. And inside, you'll be actually having the cat2 here, sir. Now, what is this cat1 and cat2 here? Cat stands for C for carnitine, A for acyl transferase. Carnitine acyl transferase 1 and carnitine acyl transferase 2, sir. Carnitine acyl transferase 1 and transferase 2, sir. That's all. By this, transport completed. Acyl CoA transported into the mitochondria. I'll pass. Make a note of this one, sir. Now, if it is done here, I'll tell you one important thing here, sir. There was a question asked in your FMG exam. Sir. See, you people have been to the, airport. You know? Now, at airport, you have seen there will be a bus which will be going to one terminal, picking the people and coming to another terminal. Then again, going to another, back to the same terminal, again picking the people and come. That bus is known as shuttle bus. That is shuttling from one terminal to another terminal. Welcome to our car here, sir. So this car, carnitine, it is coming out from the mitochondria, picking up acyl and becoming acyl carnitine, and then again going inside and dropping acyl. Then again it is going to come back. So it is actually carnitine coming outside, picking acyl, going inside, dropping acyl. Again coming outside, picking acyl, going inside. So therefore, doctors kept the name of this one as carnitine shuttle. So what is this shuttle service known as, sir? Carnitine shuttle. And once in your FMG exam, the question was carnitine shuttle operates in which pathway? So which pathway we are learning? Beta oxidation. So carnitine shuttle operates in which pathway? Yes, it is going to operate in the beta oxidation of fatty acid. And then one more question that you have to learn from here will be the cat1. Cat1 will be the rate limiting enzyme, sir. Cat1 will be the rate limiting enzyme. Now, I hope you still remember like which topic we are learning. Don't get confused here, sir. I told you in the overview very clearly. Yes, we are learning which topic here right now? Beta oxidation of fatty acid right now, sir. Okay? So in beta oxidation of fatty acid, we are first done with number one, which topic is done here, sir? Activation done. And then we are also done with the transport. Transport is also done. And now, my dear doctors, the third and the final thing, we are now going to learn about the beta oxidation proper. What we are going to learn now? Beta oxidation proper. Okay? So write down the third step right now, sir. That is beta oxidation proper. Kahan hai? Beta oxidation proper. And you know very well where it is going to take place now. It is taking place in the mitochondrial matrix here, sir. So the beta oxidation proper will take place in the mitochondrial matrix. Is it okay? And what is the substance present in the mitochondria right now? Acyl CoA, जो हमने अभी transport किया था. Now, this acyl CoA will undergo 4 reactions. Listen here very carefully, sir. Don't disturb your patience here. Let us be a little bit patient for 2-3 minutes. Okay. Acyl CoA will undergo 4 reactions. It will undergo how many reactions? 4 reactions, sir. Okay. And the good news here is that you people don't have to remember the intermediate substances. Please follow biochemistry exactly as I'm teaching you here. No need to remember the intermediate substances. Finally, what is going to happen here? The acyl CoA will actually break down to form acetyl CoA, and then there will be acyl CoA. Now, don't get confused. Why is sir telling like continuously about acetyl CoA, acyl CoA, acetyl CoA, acyl CoA? I'll make you understand everything. You just write down. Now, out of this 4 reaction, what you have to learn is the first reaction will be the oxidation reaction. Okay. The second reaction will be the hydration reaction. Okay. The third one will be again oxidation reaction. Okay. And the fourth one will be the cleavage. It is going to break down. Oxidation, hydration, oxidation, cleavage. They're like 4 reactions here. Okay. So you don't have to remember the intermediate substances. No need to learn about that one. Please don't waste your time in that one. They will never ask you in FMG exam, NEET PG, USMLE, anywhere they will never ask you. Okay. So they're like 4 reactions: oxidation, hydration, oxidation, cleavage. Now, my students are expert in oxidation. What is the meaning of oxidation? Chaliye sir, फटाफट बताइए. Tell me quickly right now, sir. What is the meaning of oxidation? Come on, everybody. Yes, very good, Dr. Vishal. Removal of hydrogen. And whatever hydrogen will be removed here will be accepted by FAD. FAD will accept the hydrogen to form FADH2. Hydration means what, sir? Addition of water. And then again, oxidation means what? Removal of hydrogen. And that will be taken up by NAD plus to form what, sir? NADH plus H plus. That's the reason why I taught you about the oxidation in the very beginning of biochemistry. Understanding here? So we don't have to like worry about that one. We know that very well. And last में क्या हो रहा है? There is a cleavage. There is a breakdown for acetyl CoA and acyl CoA. Done, sir. Are you all clear with this one here? Now, we are done with all the 3 stages right now, sir. So number 1, we are actually done with what here? Number 1, we are done with activation. Number 2, we are done with the transport. Number 3, we are done with the beta oxidation proper. And now it's only about the final conclusion. Please stop writing and look at the screen right now, sir. Please look at the screen here. Otherwise, there'll be confusion. Please look at the screen here right now. I started with fatty acid. It is like the climax of the story right now, sir. Listen carefully. I started with fatty acid, but no one asked me like which fatty acid is this one, sir. We are having so many of them: linoleic acid, linolenic acid, arachidonic acid, oleic, stearic, palmitic. मतलब बहुत सारे fatty acids होते हैं. Now, this is which fatty acid here? Remember, in human beings like you and me, the fatty acid which is metabolized will be the palmitic acid, sir. And palmitic acid will have 16 carbons in that one. How many carbon? 16 carbon. And please don't call it as palmitic acid, sir. It's not palmitic, it is palmitic acid. Okay, now L will be silent there. So now, how many carbons are there? 16 carbon. And now, my dear doctors, we are going to count right now. अब हम क्या करेंगे? काउंट करेंगे. तो मेरे साथ में रहिएगा. We cannot do it again and again. Okay? Now, so how many number of carbons do you have in this fatty acid? 16 carbon. Now, during activation, did we add any carbon? No. Did we remove any carbon? No. Because in biochemistry, we know very well, carbon is added or removed in the form of carbon dioxide. So in this reaction, are you all able to see carbon dioxide anywhere, sir? Nowhere. So therefore, how many number of carbons here in acyl CoA? 16 carbon. During transport, did we change any number of carbons? No. So we have just transported this one inside. So still how many carbons are there? 16 carbons. Now, beta oxidation proper. Acyl CoA mein kitne carbon hai, sir? 16. Now, what is the meaning of oxidation? Removal of hydrogen. Did we change carbon? No. Still 16 carbon. What is the meaning of hydration? Addition of water. What is the formula of water? H2O. H2O, did we add any carbon? No. Then oxidation means again what's the removal of hydrogen? But that means what I'm trying to tell you is that right from the very beginning till here, it is still having 16 carbons. And now, my dear friends, welcome to the last step here. There will be cleavage. That 16 carbon chain will actually break down and 2 carbons will be removed off. And that 2 carbons will be removed in the form of acetyl-CoA. And we are now remaining with how many carbons? Sir? 14. And even 14 carbon means many carbon, na? That is also acyl-CoA. Okay. अब इस acyl-CoA का क्या होगा? What will happen to this acyl-CoA? That acyl-CoA will again go back here. And then what is going to happen? That will undergo oxidation, hydration, oxidation, cleavage. And then again, what is going to happen? 2 carbons will separate apart and remaining with 12 carbons. The 12 carbons will again go back to acyl-CoA. So again, the thing will keep on continuing. Beta oxidation proper bar hota jayega until and unless the entire 16 carbon will break down to 2 carbon acyl-CoA. Did you understand this one? Come on, my dear friends. Did you understand this one? So ये बार hota jayega. Therefore, my dear friends, what is the final end product of beta oxidation of fatty acid? Only acetyl-CoA. That is what we have written in the overview. Overview में वही तो लिखा था ना हमने, है ना? Let me take you back to the overview. हाँ, यहाँ देखो आप. What is the starting substance? Fatty acid. What is the end product here? That is your acetyl-CoA. ठीक है ना? So what is the final end product here? That is acetyl-CoA. समझ में आया? Did you understand? Did you all understand? Yes, very good, sir. So in this manner, what is going to happen? We are going to begin from fatty acid, and finally, what is the end product? The end product will be acetyl-CoA. So therefore, now write down note करके लिखो, sir. Note number 1. First of all, what is the rate limiting enzyme of beta oxidation of fatty acid? That is CAT1. You have to remember this point for your exam, sir. Is it okay? CAT1. CAT1 is the rate limiting enzyme here. Number 2, what is the net gain of ATP, sir? The net gain of ATP in beta oxidation proper will be 106 ATP, sir. 106 ATPs when you're starting from palmitic acid. और मैंने आपको ऑलरेडी कहा था palmitic acid will have how many carbons, sir? 16 carbon. कितने कार्बन होते हैं? 16 carbon होते हैं. So net gain of ATP will be 106 ATPs. So you can see like we will be getting lots and lots of energy from the fatty acid. Then note number 3, on one side you write down there as fatty acid. On the other side you write down there as the end product. Now, suppose if there is an even chain fatty acid, what is the end product, sir? Acetyl-CoA. Hello, my dear doctors. अभी हमने start किया था with the palmitic acid. Can you please answer me in the chat box? Palmitic acid, how many carbons are there, sir? How many carbons are there in the palmitic acid? 16 carbons. Very good. So 16 is even or odd? Even chain. So even chain fatty acid, the end product will be what? Acetyl-CoA. Okay. Now, if it is odd chain fatty acid, sir, the end product will be propionyl-CoA. और ये हमने अभी break से पहले पढ़ा था. Just before the break, we have learned about this one in vomit. आपको याद है vomit path पे? And you can turn the page back and see there in the vomit, O stands for odd chain fatty acid. So the end product of odd chain fatty acid will be propionyl-CoA. So these two things should be like very, very clear in your mind. Even chain fatty acid end product will be acetyl-CoA. Odd chain fatty acid product will be propionyl-CoA. So these are all the points that you have to remember for your FMG exams. So what is the rate limiting enzyme? CAT1, and the net gain of ATP will be 106 ATPs, and then the end product, sir. Is it okay? So अगर even chain होगा acetyl-CoA, odd chain होगा तो propionyl-CoA. Done, sir. This topic is done, sir. The number of ATP will be 106 ATP, sir. You don't have to worry about how to calculate and all. Just remember that number. There is a big method of calculation for that one. Big method. So basically, you just remember 106 ATP, sir. It is glutathione peroxidase. Glutathione oxidase nahi hota hai. It is actually glutathione peroxidase. Doctor. So now going back to our overview here for the last and final time, my dear friends. we are actually done with beta oxidation of fatty acid हो गया. And now welcome to ketogenesis. Yesterday someone was asking, right, what is this ketone bodies and all? I think Najmuddin was the one who was asking me yesterday, sir, ketone bodies kya hote hain and all. Welcome here, Najmuddin. So now we are actually going to learn about the ketogenesis, sir. But before learning about the formation of ketone bodies, I have observed that many of the students don't even know like what are ketone bodies, sir. First of all, let us learn कि what are the ketone bodies. Ketone bodies होते क्या है? Remember, there are three ketone bodies, sir. Number one, you'll be having acetoacetate. Number two, there will be acetone. And number three, you'll be having beta hydroxybutyrate. पहले तो इनके नाम याद करो सर. First, learn the names right now, sir. Okay, now. So there are like three ketone bodies. Number one, you'll be having acetoacetate. Number two, you'll be having acetone. And number three, there will be beta hydroxybutyrate. There will be three ketone bodies. So you'll be coming across these ketone bodies in many subjects there, sir. Ketosis, even in diabetes also, diabetic ketoacidosis, DK. So you'll be coming across this one in many subjects. So remember, what are ketone bodies? Pehle wo seekho dhyan se. Number one, acetoacetate. Number two, acetone and beta hydroxybutyrate. Now, after learning the names of the ketone bodies, now we are going to start with ketogenesis. Ketogenesis begins with acetyl-CoA. वो तो हमको overview से पता है. Already we know from the overview. Acetyl-CoA combines with another molecule of acetyl-CoA. Now these two will combine to form acetoacetyl-CoA. Acetyl-CoA, acetyl-CoA, acetoacetyl-CoA. Then that will combine with one more molecule of acetyl-CoA to form HMG-CoA. ये हमने क्या किया सर? जोड़ दिया. We have joined them together. Acetyl-CoA with acetyl-CoA, acetoacetyl-CoA. Add one more molecule of acetyl-CoA, it is going to become HMG-CoA. And now इसको break कर दो sir. From this, remove one molecule of acetyl-CoA. Oh my God! Now you're going to kill me. यहाँ से एक acetyl-CoA निकाल दो. So then what is remaining? We are going to remain with acetoacetate. What is that one? Acetoacetate. मेरी नजर से देखो, we got the first ketone body. And this ketone body itself is going to form the remaining two ketone bodies. That is acetone. Another one will be beta hydroxybutyrate. ठीक है बच्चों, that's all. मैंने क्या कहा था आपको स्टार्टिंग में? ट्रस्ट मी, बिलीव मी, 2 मिनट में हो जाएगा. हो गया सर, कीटोजेनेसिस हो गया. तो पहले जोड़ दिया जाए और फिर तोड़ दिया जाए. फर्स्ट यू जॉइन देम टुगेदर, acetyl-CoA, 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 and then you break them. खत्म. Is it okay? So therefore, I think now you also have got a little bit of experience कि यार सवाल कहाँ से आ सकता है. Now you only tell me, will they ask any question from here? Amin. Will they ask any question from this pathway, sir? ये तो टाइम पास पाथवे, यहाँ तो बस जोड़ना और तोड़ना ही है. तो अब मैं आपको बताऊंगा. नाउ आई विल टेल यू कि सवाल कहां से आएगा. द क्वेश्चन विल कम फ्रॉम वेयर. क्वेश्चन नंबर 1. Out of the three ketone bodies, which is the primary ketone body? The primary ketone body will be acetoacetate. This is the primary one, the main one. Number one question. Number two, where is this ketogenesis taking place? Location, of course, liver. बायोकेमिस्ट्री में अगर कुछ याद नहीं आ रहा है तो कहाँ होता है? लिवर में होता है. लिवर. Inside the liver, it will be taking place in mitochondria. Previous year question in FMG. In the liver, it will be taking place in the liver cells, mitochondria. Then number 3, yes, what is the use of these ketone bodies? Some students were asking me, what is the use of these ketone bodies? So my dear friends, remember ketone bodies are a very good source of energy. Najmuddin, ketone bodies are a very good source of energy. Don't think that ketone bodies are going to be formed at the end. तो इसीलिए इसका कुछ काम नहीं होगा हमारी बॉडी में ना, ऐसे मत सोचिए सर. रिमेंबर ketone bodies are very important, especially during starvation. They are very good source of energy. Okay. And during starvation, what will happen? Like normally मैंने आपको कल बताया था ना in glucose transporter कि brain requires glucose all the time. It requires glucose at any cost. अब starvation मतलब you are not at all eating almost up to 3 days. Okay, now almost up to 3 days, like 72 hours. So at that time, what is going to happen? Now we are not getting glucose. है ना, ग्लूकोज नहीं मिल रहा है. तो नाउ एट दैट टाइम इवन ब्रेन ऑर्गन लाइक ब्रेन विल आल्सो स्टार्ट डिपेंडिंग ऑन कीटोन बॉडीज ओनली. क्या करेंगे, है ना, एनर्जी तो चाहिए कहीं ना कहीं से. तो देयरफॉर माय डियर फ्रेंड्स रिमेंबर कि ब्रेन भी विल आल्सो यूटिलाइज द कीटोन बॉडीज. लाइक many organs in our body will start utilizing the ketone bodies as a source of energy at the time of starvation. So starvation में, fasting में क्या होता है, ketone bodies will be acting like a source of energy, sir. But सवाल वो नहीं है. The question is not about which organs will use ketone bodies. The question is which organ cannot use ketone bodies. बहुत सारे organs use करते हैं, brain, skeletal muscle, heart, everyone will use the ketone bodies. But which organ cannot utilize the ketone bodies? So which organ cannot utilize the ketone bodies, sir? Number one, the liver, and number two, your RBC, sir. अरे which are a liver? लिवर खुद कीटोन बॉडीज फॉर्म करता है लेकिन वो यूटिलाइज नहीं कर पाता है. तो प्लीज रिमेंबर लाइक दैट सर, लिवर इटसेल्फ इज गोइंग टू फॉर्म द कीटोन बॉडीज बट इट इज नॉट एबल टू यूटिलाइज द कीटोन बॉडीज. आर यू ऑल वेरी क्लियर विद दिस वन हियर? Yes, my dear friends. Okay, so in this manner I want you to remember these 3 points here for the sake of your exams. हो गया. By this we are done with what here, sir, about the fatty acid. तो synthesis of fatty acid भी हो गया और breakdown of fatty acid भी हो गया. Are you all clear with this one here? भाई साहब आपको कुछ याद है कि भूल गए? Do you all remember what are we learning? Lipid metabolism topic we divide into how many categories, sir? 3 categories here. Number 1, cholesterol synthesis degradation done, and number 2, fatty acid synthesis and degradation done, sir. ठीक है ना? तो ये दोनों टॉपिक भी हो गया सर, कोलेस्ट्रॉल भी हो गया and then we are done with the fatty acid synthesis as well as degradation. Now the last and final topic that is remaining is lipoproteins. Acetyl CoA carboxylase. नजमुद्दीन हमने अभी पढ़ा था ना यार, तुम कहाँ थे? ये वाला. The third one which utilizing biotin will be acetyl CoA carboxylase. Sure. Any more doubts? Now the next thing that we are going to learn is the lipoproteins. ठीक है ना? So in the lipid metabolism, we are done with the cholesterol and we are done with the fatty acid. These 2 things are done, sir. And now the last and final thing that is remaining is lipoproteins only, sir. And lipoproteins तो ऐसे हो जाएगा सर. कैसे हो जाएगा? ऐसे. ठीक है ना? 10-15 minutes, this topic will be done, sir. Please write down the heading there as lipoproteins. This is a beautiful conceptual topic. From the name itself, we can understand that lipoproteins are made up of 2 things, sir. Number 1, lipids, and number 2, proteins. And the proteins here are given a special name. They are known as apoproteins, sir. Lipoproteins are made up of 2 things. One is the lipid, another one is the protein. And this protein is given a special name here that is known as apoproteins. Now, what is this lipid part made up of, sir? Remember, the lipid part is made up of triglycerides and cholesterol. And this apoproteins will be of different varieties. You'll be having apoprotein A, then there will be apoprotein B48, there will be apoprotein B100, there will be apoprotein C, and then you'll be having the apoprotein E. So lipoproteins are made up of 2 things, sir. One is your lipids, another one will be the proteins, sir. Okay, na? So lipids are made up of triglyceride and cholesterol, and proteins are made up of what, sir? Apoprotein. ठीक है? So now what is going to happen here? Whenever you're going to combine them together it is known as lipoproteins. ठीक है? It is known as lipoproteins. Introduction हो गया sir. Now once you have understood the introduction here, now please write down the heading there as types. How many types of lipoproteins do we have, sir? Number one, there will be chylomicron. Number two, very low density lipoprotein, intermediate density lipoprotein, low density lipoprotein, and then you'll be having high density lipoprotein. So you're having 5 different types of lipoproteins. I repeat again, chylomicron, VLDL, IDL, LDL, HDL. Okay, Ji? Are you done? Now, once you are done with this introduction here, sir, now we are going to make this topic easy just by 2 diagrams here. बस 2 डायग्राम और ये टॉपिक कॉन्सेप्चुअली खत्म हो जाएगा सर. कॉन्सेप्चुअली विल बी डन सर. अब पहले अपना प्लानिंग सुनो ध्यान से सर. प्लानिंग बहुत इंपॉर्टेंट है. प्लानिंग इज ऑलवेज इंपॉर्टेंट, है ना? लिसन वेरी कॉन्सेप्चुअली हियर. In that 2 diagram, in the very first diagram, what we are going to do is, in the first diagram, we are going to learn about the exogenous fat. Now, what do you mean by exogenous fat, sir? The fat which you are consuming in your diet. ठीक है ना? So exogenous fat, I can also call it as dietary fat, the fat that you are going to consume in your diet, sir, from outside. है ना? और आजकल तो बड़ा कॉमन है, all the junk food that you are eating, you know, burger, pizza, cheese, mayonnaise, पता नहीं क्या क्या. All that will be actually having your dietary fat, the fat that is taken from outside the body. Now, second diagram which I am going to tell you is actually about that is your endogenous fat, the fat that is produced inside of a body, जो हमारी body के अंदर produce होता है सर. So in these 2 diagrams, I am going to finish off the lipoproteins topic, sir. Lipoprotein हो जाएगा. So one is about the exogenous fat, another one is about the endogenous fats. Exogenous fat and endogenous fats. Are you ready now? Are you ready now, sir? The first, you write down the heading there as metabolism of chylomicron. Metabolism of chylomicron, sir. Now imagine this is your intestine here, sir. मान लो कि ये वाला आपका intestine है, sir. Now try to understand my planning. On day 1, I taught you we have consumed carbohydrate rich diet. Yesterday we have consumed protein rich diet, chicken, mutton and all. And today we are going to consume the fats, the lipids rich diet, है ना? तो अब यार ये कहाँ होता है? तो I think you people have been to McD, McChicken, Burger, है ना? KFC and all. हाँ, Burger King गए होंगे ना? तो वहां क्या होता है? The moment you go there, what will happen, you know? कि वहां पर ना at the counter there will be one beautiful girl standing, and boys knows this very well कि जैसे ही आप वहां पर जाते हो, the moment you look at that beautiful girl there, खत्म खत्म. You will just keep on giving, you will just keep on seeing her, और आप उसको देखते देखते ना सारे ऑर्डर देते जाते हो, है कि नहीं सर? तो the moment you go to McD, she'll be asking the first thing, sir, what do you want to have? Okay. तो मन में तो कुछ और ही चलता है कि what I want to have, लेकिन मतलब दिल पर पत्थर रखकर बताना पड़ता है, I just want to have Mac chicken burger. And she'll be like small or large. By small से अपना क्या होगा? Large. उसके बाद में तीसरा सवाल आता है ना? हाँ कि सर डू यू वांट एक्स्ट्रा चीज लेयर? लगा दे, जो लगाना है लगा दे, तू भी आ जा. So what will happen, now you'll be getting that Mac chicken burger with that extra yellow colored cheese layer. And यार आप लोग बर्गर को कैसे देखते हो मुझे नहीं पता है. एज ए बायोकेमिस्ट्री टीचर मुझे ना बर्गर में ना पूरी बायोकेमिस्ट्री नजर आती है. तो in the burger what will happen, there will be one क्या कहते हैं उसको, ब्रेड, ब्रेड नहीं, हाँ ऊपर बन, बन बोलते हैं, हाँ ऊपर एक बन होता है और नीचे एक बन होता है, दैट इज कार्बोहाइड्रेट. एंड देन वी हैव ऑर्डर्ड अ मैक चिकन बर्गर. तो मैक चिकन बर्गर में क्या होता है सर, वहां पर ना वो चिकन होता है, चिकन 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 पैटी होता है. एंड देन वी हैव ऑर्डर्ड दैट विद एक्स्ट्रा चीज लेयर. ओए होए होए होए, यहाँ पर ना येलो कलर चीज लेयर, ओ माय गॉड, ऐसे ना मेल्ट होते रहता है. तो ऊपर और नीचे जो बन होता है ना, दैट इज लाइक कार्बोहाइड्रेट, दैट चिकन इन द मिडिल विल बी द प्रोटीन, एंड देन दैट एक्स्ट्रा चीज लेयर विल बी योर फैट वाला लेयर. हाँ हाँ, तो कार्बोहाइड्रेट, प्रोटीन एंड द लिपिड वेल बैलेंस्ड, ऑल द थ्री ऑफ देम इन द बर्गर. और आज मान लो कि हम ये ना बर्गर कंज्यूम कर रहे हैं, वी आर कंज्यूमिंग दिस बर्गर टुडे, वी आर ईटिंग दिस बर्गर नाउ. और आज आपको मैं बताऊंगा कि वो एक्स्ट्रा चीज लेयर का क्या होता है, व्हाट विल हैपन विद दैट एक्स्ट्रा चीज लेयर टुडे. अब यार ये बताओ, दिस इज द फैट यू आर कंज्यूमिंग इन योर डाइट ना, तो कैन आई कॉल इट एज डाइटरी फैट? Yes, it is dietary fat. And this dietary fat we are taking from outside now. So therefore, I can also call it as exogenous fat. चलो, कहानी कहानी सुनो सर, मस्त कहानी. आई एम वेरी गुड एट स्टोरी टेलिंग. तो हमने क्या किया सर, मैक चिकन बर्गर विद एक्स्ट्रा चीज लेयर खा लिया. एंड आफ्टर ईटिंग दैट, दैट फैट लेयर इज गोइंग टू योर स्टमक एंड इंटेस्टाइन. और इंटेस्टाइन से यहां से क्या होता है, इट विल बी अब्सॉर्ब्ड. नाउ after absorption, first of all, there will be triglyceride and cholesterol. And triglyceride and cholesterol, that is going to form which part, sir? The lipid part. Lipids will be yellow in color, so I'm just highlighting this with a yellow color here. And attached to this, you'll be having protein. Which protein? Apoprotein B48, apoprotein E, and then there will be apoprotein C. Now, my dear friends, look at this molecule here. Now, this substance here, is it having the lipid part? Yes, it is having the lipid part. Is it having the protein part? Yes, it is having the protein parts also, every protein. Now, lipid plus protein, it is a lipoprotein, and this one will be the chylomicron. यही तो है chylomicron. Chylomicron ऐसे नजर आता है. You'll be having the lipid part and the protein parts. And to be very frank with you people, actually ना chylomicron यहीं पर फॉर्म हो जाता है. कहाँ फॉर्म होता है? इंटेस्टाइन में. So it is forming in intestine only here, sir. But I am like, why to draw it like two times करके? I am drawing only one time there, sir. So from today onwards, the first thing that you have to remember is that whenever you're consuming this, all this oily foods, you know, junk foods, जो होता है ना extra cheese layer and all, वो हमारे intestine में क्या बन जाएगा? Chylomicron बन जाएगा. It is going to become what, sir? Chylomicron. And that chylomicron will be absorbed. Now, after absorption, अब ये chylomicron कहाँ है, sir? So remember, this chylomicron is right now present in our blood. It is nicely circulating in the blood. Now, while circulating in the blood, this chylomicron will come in contact with the peripheral tissues. So it is coming in contact with what, sir? Some peripheral tissues here, sir. And one of the best example for the peripheral tissues that we can take here will be adipocytes, sir. I am taking the example of adipose. It is going to come in contact with which cells? Fat storing cells, adipocytes. It will be like easy for us to understand, ना, fat storing cells, क्योंकि वो fat store करते हैं ना. So it is coming in contact with adipocytes. But listen here very carefully with the 200% concentration here, sir. बस यहीं पर आपको सीखना है. If you understand this point here, just 2 points here, sir. अगर ये समझ गया तो टॉपिक खत्म, सर, टॉपिक खत्म. अब मेरा सवाल यहां पर ये है, my question is that chylomicron is going in our blood, and in our body we are having like many cells, many cells, so millions and billions of cells. How is chylomicron able to identify adipocyte only? We want to understand in depth right now. How is it able to identify the adipocytes only, sir? So remember, on the surface of the adipocyte, you'll be actually having special receptors for chylomicron. God has given special receptor for the chylomicron. Okay, ji, ठीक है. But now I am like asking in more depth right now, sir. Sir, you said this receptor is present there. You are saying that the receptor is present over there, but How is chylomicron able to identify that receptor? Chylomicron receptor को कैसे identify करता है? So whatever apoprotein B48 and whatever apoprotein E is present here, these proteins here, they are not just for decoration here, sir. They are not just for, fancy decoration here. They have some function. They have some responsibility, and they are the one which are going to identify this receptor. क्या बात है. So they'll identify the receptor, and the chylomicron will go and get attachment. Attachment हो गया. Then, sir, what is the function of this ApoC then? This ApoC will go here and it will going to activate an enzyme. Which enzyme it is going to activate? It is going to activate an enzyme known as LPL. And जैसे ये LPL activate होता है तो क्या होता है? What will happen? To understand what will happen, first you have to understand the full name. What is the full name of LPL? Full form? Lipoprotein lipase. What is the full name of LPL, sir? Lipoprotein lipase. And here we have been learning biochemistry since 3 days. ये तो आपको अच्छे से पता है. You know that very well right now. So what is the meaning of lipase? Lipase breakdown. Breakdown of what? Lipid. But which lipid is going to break down? Breakdown of lipids only. But which lipid is going to break down? So remember, lipases are going to break down the lipoprotein. कौन सा वाला है lipoprotein right here? Chylomicron. So apoprotein C will go and activate this enzyme. This enzyme will in turn break down the fat present in the chylomicron. ये fat को break करता है सर. Lipoprotein lipase. सब कुछ नाम में ही लिखा है. Lipoprotein lipase. It is going to break down the lipid. And once it is going to break down that lipid, sir, into small, lipid globules, and that small lipid globules will now happily enter inside the adipocyte. ये adipocyte में store हो जाते हैं. Wow. अब समझ में आ रहा है? Are you getting a point here right now? That will be stored in the adipocytes. And yes, आपको पता है ये कहानी कहाँ से शुरू हुई थी? मैगडी से शुरू हुई थी, है ना? तो चलिए कोरिलेट करते हैं. Let us correlate right now, sir. So one fine morning you went to McD. In McD you ordered a McChicken burger, extra large McChicken burger with additional cheese layer. That cheese layer will come into your stomach and intestine, and it is going to become chylomicron. Chylomicron will be absorbed, and after absorption, it is coming in the blood. And in the blood, what is going to happen? Apoprotein B48 and E will identify the receptor, and C is going to activate this enzyme. And once that enzyme is activated, that fat will be actually stored in the adipocytes, and we keep on becoming like fat. तो हम जंक फूड खाते जाते हैं, ऑयली फूड खाते जाते हैं, और ऐसे ना मोटे मोटे होते जाते हैं. समझ में आ रहा है? Are you understanding me? So from today onwards, whenever you eating all these junk foods and all, please remember this diagram here. से ये diagram याद रखिए आप. ठीक है ना? This is the culprit. Chylomicron मस्त में आपके फैट को ले जाकर वहां पर एडिपोसाइट में स्टोर कर देता है. हाँ, शवरमा, फलाफिल सब खाओ. अच्छा, now will the entire fat be stored in the adipocyte? क्या वो पूरा वाला फैट स्टोर हो जाता है यहाँ पर? ना. All the fat will not be stored there, sir. All the fat will not be stored there. Little bit of fat will be remaining, and that remaining fat will be, yes, triglyceride and cholesterol will be along with apoprotein B48 and apoprotein E. Kani apoprotein C undadu. There will be no apoprotein C. Why? Because apoprotein C will be used for activation of that enzyme. It is used for that activation of the enzyme, तो वो नहीं होता है. It will not be there. Okay. Now this is the remaining molecule of chylomicron. That is why doctors have kept a beautiful name for this one. This one is now known as chylomicron remnant. Remaining molecule of chylomicron, chylomicron remnant. अब इसका क्या होगा? What will happen to the chylomicron remnant? लिवर कहता है भाई साहब आपका काम हो गया, आपने मस्त में फैट डिपॉजिट कर दिया, आ जाओ वापस, काम हो गया, आपने उसका काम पहचान लिया. ठीक है? So now this chylomicron remnant will return back to the liver, Why? Because liver is the centralmost organ for fat metabolism. So again, it is going to return back to the liver. So done. तो आपने जो भी ऑयली फूड खाया है, whatever junk food that you have consumed, chylomicron will store that fat in the adipocytes and come back to the liver. It's a beautiful diagram. And that is about our first diagram here. So that is metabolism of chylomicron, exogenous fat, the fat from outside the body. Yes. Any doubts? Let me know, sir. I'll give you 1 minute of time. Should we know the name of the receptor, sir? The name of the receptor is chylomicron receptor only, ma'am. It is nothing special. Chylomicron receptor. On the surface of adipocyte, we'll be having chylomicron receptor. Now, मैंने आपको starting में क्या promise किया था, sir? What I promised you in the very beginning? I'm going to give you only two diagrams here. One diagram, exogenous fat. And now, one more diagram is about the endogenous fat, sir. The fat that is produced in our body. And the good news here is that the next diagram that I am going to teach you, my dear friend. ठीक है ना? The diagram is like copy paste here. तो ना मुझे पढ़ाना है ना आपको पढ़ना है. Neither I have to teach you nor you have to learn, sir. Let us just do the copy paste right now. Let's start your favorite work. Copy paste. Now, my dear friends, write down the heading there as metabolism of VLDL, very low density lipoprotein. Metabolism of VLDL. Sir. Liver actually has one of the function that it synthesizes VLDL, sir. Liver synthesizes VLDL. The liver has got like many functions, sir. And one of the function of the liver is that it synthesizes VLDL. Now tell me, if liver is synthesizing VLDL, so this one is like exogenous fat or endogenous fat, sir? Endogenous fat. After synthesizing VLDL, liver will transport that VLDL into the blood. VLDL will consist of triglyceride and cholesterol. And triglyceride and cholesterol, this is going to form the lipid part. I'm just highlighting with yellow color because fats or lipids will be yellowish in color. Attached to that, there will be apoprotein B100, and then there will be apoprotein E. And then you'll be having the apoprotein C. Now, please look at this structure here very carefully. You're having lipids here. So therefore, lipids will be there as well as the protein is there. So it is a lipoprotein. Which lipoprotein is this one? This one is VLDL. So VLDL will be appearing like this here. So अब क्या होगा? Now what is going to happen? VLDL is going to identify the receptor. How it is going to identify the receptor? Apoprotein B100 and apoprotein E will identify the receptor. Then what is the function of ApoC? ApoC will actually go and activate the enzyme known as it is going to activate the enzyme known as LPL, lipoprotein lipase. And once this enzyme lipoprotein lipase is activated, you know very well the full name of that one. It is going to break down the lipids of VLDL and store that lead lipids into the adipocytes. Sir. Wow. Same thing. I told you it is like copy paste only, sir. And will be like once more, it will be like a revision here. Then will the entire lipids be stored here? No, entire lipids will not be stored here. Will be left out with a little bit of triglyceride and will be left out with little bit of cholesterol. Attached to that, there will be apoprotein B100 and then you'll be having the apoprotein E. But apoprotein C will not be there, sir. Why? Because C will be used for activation of enzyme. Now, what is the name of this molecule right now, sir? This is the remnant of VLDL. So therefore, VLDL remnant. And VLDL remnant is also known as IDL, intermediate density. So that itself is known as intermediate density lipoprotein. Ab iska kya hoga? What will happen to this one? This is again taken back by the liver, sir. Same story, sir. Same thing. And yes, Dr. Shinit, it will be again what is going to happen with the remnants there. They'll be again recycled inside the liver. ये दोबारा liver में use हो जाते हैं. That is the reason why Dr. Shinit, you see, whenever you are actually having this oily food or junk food and all, and interfering with the metabolism of the fat, which God has naturally given us, है ना? Always as a human being will be interfering with the nature only, है ना? तो सब लोग जो नेचुरल चीजें हमको ऊपर वाले ने दी है, हम उसमें गड़बड़ी करते रहते हैं, है ना? तो बिकॉज ऑफ दैट इंटरफेरेंस इन द फैट मेटाबॉलिज्म, ऊपर वाले ने कहा है कि मतलब तरीके से खाना खाओ भाई, दुनिया आपके लिए बनाई है, तरीके से खाओ, जितना लगता है उतना खाओ। नहीं, हमको तो मतलब है ना, जंक फूड, दिस फूड, 4 o'clock स्नैक्स, चिकन पकोड़ा, डीप फ्राइड चिकन, दिस दैट एंड एवरीथिंग वी हैव टू, है ना, फ्रेंच फ्राइज, ओ हो पेरी पेरी लगा के, तो हम खाते जाते हैं, फैट ज्यादा हो जाता है। तो जैसे आप इंटरफेयर करोगे तो क्या होता है डॉक्टर शनील, व्हाट इज गोइंग टू हैपन, द फर्स्ट ऑर्गन टू गेट अफेक्टेड देयर विल बी योर लिवर। इसीलिए देखते हो ना, फैटी लिवर बड़ा कॉमन होता है। whenever you are interfering with the fat metabolism, eating oily food, junk food and all, fatty liver होता है because of that, this thing only, because liver is the main organ। अब उसके ऊपर अगर आप ओवरलोड करोगे तो ऑब्वियसली ये चीजें होगी ना। ऐसे। चलिए सर, वॉट एवर इट इज, लेट अस गो अहेड राइट नाउ। इंटरमीडिएट डेंसिटी लिपोप्रोटीन है वो, ठीक है ना। Now, once you are done with these two here, hello my dear friends, are we all done with the chylomicron? Chylomicron हो गया है sir? Yes. Are we are done with VLDL? Yes. Are we done with IDL? Yes. Now what is left out? We are left out with low density and high density. I'll tell you one method how to remember this one. Yeah, देखो. Now this one here will be the intermediate density lipoprotein, sir. Intermediate density. From here, I am going to remove one more protein. Apoprotein E हमने निकाल दिया. Whatever. I am removing like one more protein from here. Now, if you remove like one more protein from here, density बढ़ेगा कि कम होगा? Density will increase or decrease? Of course, the density will further decrease, sir, because density depends on the percentage of protein. Percentage of the protein, sir. Density क्या होगा? Further decrease होगा. Density is going to decrease further, sir. Now, already, what is the name of this one here, sir? इसका नाम क्या है? The name of this one is actually the intermediate density lipoprotein, sir. So now, if you remove like one more protein, the density will decrease. So from the intermediate density lipoprotein, it is going to become what, sir? The low density lipoprotein. Are you understanding me now? So it is no more intermediate. From there, it is going to become low. तो इसको याद रखने का तरीका ऐसा होता है. This is the way how you have to remember this one, sir. So therefore, welcome here, sir. Triglyceride and cholesterol. Along with that, there will be apoprotein B100 only. This one will be known as what, sir? Low density lipoprotein. LDL. That's all. From intermediate, you remove one more protein, it becomes low density. So that is an easy way to remember. And the last and final, which is remaining right now, is your HDL, sir. HDL, again, the same thing. Lipid part will have triglyceride and cholesterol. So it's a, guys, you don't even remember this one, so lipid part. Lipid is always the same. Your focus should be only on the apoprotein, so that is changing. So in HDL, that will be attached with what, sir? Till now, we did not use apoprotein A, then there will be apoprotein E, and then you'll be having the apoprotein C, sir. Done and done, sir. Okay, that's your HDL. That is HDL, sir. Okay, done. That's all. So in two diagrams, we are done with everything, sir. Exogenous fat and endogenous fat. Fat taken from outside and fat produced inside our body, sir. Okay, now, once you are done with this one, let us just now conclude this topic, sir. Make a table. First, we are going to write down the name of the lipoprotein, and then which apoproteins are present in that one, and what about the lipid part, sir. First one will be your chylomicron. Come on, sir. I'm not going to teach you now again and again. मैंने आपको already पढ़ा दिया है. Look at the two diagram. You'll be able to answer everything, sir. Look at the two diagrams there. You'll be able to answer everything. So diagram देखकर बताओ. In the chylomicron, which apoprotein do you have? Apoprotein B48, apoprotein E, and then C. Good. Very nice. Very nice. VLDL, apoprotein B100, then E and C, sir. क्या बात है, very nice. Intermediate density lipoprotein, there will be apoprotein B100 and E. Why? Because intermediate is nothing but what's a remnant of VLDL only. Now from here, even remove the E also, it is going to become your LDL, apoprotein B100 also. Then finally, HDL will be having apoprotein A, comma E and C. Wow. Now, other students who are actually not attending our class here, sir, what they'll be doing now? They'll be actually mugging up all these things. Now, you only tell me, if you mug up all these things, will you be able to remember for a long time? No. You will not be able to remember this for the long term, sir. So, इसका ना पढ़ने का एक तरीका होता है, तो learn in this manner here, sir. Now, after this one, now tell me about the lipid part, sir. Lipid will have only and only two things only, now. One is triglyceride, another one is a cholesterol, sir. Lipid is always made up of what only? All of them will be having triglyceride and cholesterol only. So no need to write down that again and again, triglyceride, cholesterol, triglyceride, cholesterol. All of them will be having the same thing only, sir. Triglyceride, cholesterol only. But you have to remember, out of all of them, which is having the highest amount of the triglycerides and which one will be having the highest amount of the cholesterol. So remember, the highest amount of triglyceride will be present in chylomicron, and the highest amount of cholesterol will be present in LDL. Highest amount of triglyceride will be in the chylomicron, whereas highest amount of the cholesterol will be in LDL. Okay, these 2 points you have to remember. Easy. And the last and final thing in this topic is we have to remember the functions of apoproteins. Apoprotein B48, apoprotein B100, and then apoprotein E. All the three of them, their function is interaction of lipoproteins with receptor. Apoprotein B48, B100, and E. These three will be helping in what, sir? Interaction of the lipoprotein with the receptor, sir. Their function is this one. Whereas apoprotein C, iska kaam kya hai? This is helping in activation of which enzyme? LPL. And whereas the last and final one, ApoA, it is also helping in activation of an enzyme. And activation of which enzyme, sir? LCAT. LCAT. Don't have to worry about that enzyme for your FMG exam. Just write it and leave it, sir. It is also helping in activation of an enzyme known as LCAT. Are you getting me? That's all. So what I'm trying to teach you here, are you understanding, sir? Actually, लोग क्या करते हैं ना, उल्टे तरीके से पढ़ते हैं. They'll be learning the functions first. They'll be learning all this, tables first, mugging up, and something they'll be doing. To learn conceptually here, sir. If you just remember my two diagrams here, this diagram and this diagram here, automatically you'll be able to answer anything, sir. और वो तो मैंने आपको ऐसे आसान बना दिया, sir. I have made it easy for you. Is it okay? So just with these two diagrams, you'll be easily able to recall everything with guys. Are you understanding? And by this, hello, my dear friends. Welcome here. कहाँ से start किया था हमने? Lipid metabolism, cholesterol synthesis and degradation, fatty acid synthesis and degradation, and then we are done with the lipoproteins. All that VLDL, HDL, and all those things are done. Are you clear? And by this, we are done with the lipid metabolism, sir. Ajmal is telling, sir, in according to the name, VLDL is supposed to have lesser apoprotein compared to LDL. Ajmal, did you concentrate on what did I tell you properly there? I told you percentage of protein, not the number of protein. थोड़ा सोचिए उसके बारे में. You think about that one. Ajmal, you are comparing this VLDL with LDL, है ना? Very low density and low density. So you are telling that very low density में the protein should be more or less. I am not telling the number. I am not telling the type. How many types are there? I am telling the amount, percentage. यहाँ पर only apoprotein B100 है. There's only evaporating B100, but that is in more percentage. Percentage उसका ज़्यादा है when compared to these three types also. Types अलग है, percentage अलग है. तो इसलिए मैंने क्या कहा था? Density depends on percentage, not the types. I did not tell you types of the protein there. Percentage, amount. You got the clarity? Thank you. Welcome, Nico. Okay, ji, done. And by this, my dear friends, we are done with what here, sir? Lipid metabolism ho gaya, sir. So now I hope you are getting a overview, a clear idea that carbohydrate metabolism is a very big topic. Protein metabolism is like moderate one, and lipid metabolism though in 2 hours it is done, sir. Exactly. I started at 5:30 and 7:30 it is done, sir. समझ में आ रहा है आपको?